So hello students, welcome to Vedantu NEET English. This is Basavaraj, your biology master teacher. Quickly let me know in the chat right now if I am audible and visible to each and every one of you. Quickly let me know in the chat, students. Now, this is going to be a one-shot lecture on the chapter, your biochemistry one, that is biomolecules chapter, right? Now students, who can watch this lecture? Any student who do not have any understanding of this chapter, like legit zero knowledge about this chapter, can watch it. If you watch the lecture till the very end, you will understand every single concept given your NCERT. That is my guarantee. Now, if you are a student who has learned this chapter somewhere before in coaching centers or from me or any other YouTube platform, if you just listen to my lecture, you will understand completely 100% of it. And let me tell you students, after this class, your entire backlog of biomolecules can be completely cancelled from your entire study schedule. So with that being said, students, can we start today's class? If we can, show some energy in the chat, students. Show some energy in the chat. Show some fire emoji in the chat right now if we can start today's class. Because it is going to be a very long lecture, let me tell you, because it's a massive chapter. This chapter has a connection to the other chapters in your class 12th, chemistry. It could be your biology class chapter, molecular basis. So if you make your foundations right, and if you learn this chapter in detail, your life is going to be easy. Okay. So that is my guarantee from my side to you. Now it is up to you students, so to smash the like button first of all by marking your attendance, every single one of you. After that, I want every single student to watch the lecture till the very end. Promise me students, promise me in the chat, all of you. Promise me in the chat, you will watch the lecture till the very end, because that is when the true part is. Because I know, students, tell me, how many of you find it difficult? How many of you find it difficult to understand what is inhibitors? What is a cofactor? How does the enzyme reaction occurs? What is Vmax? What is half Vmax? How many of you find it difficult? Raise your hands right now. Because if you do, I'm going to make all those concepts easy for you today. Okay, I'm going to make it easy for you. Okay, so students, without any delay, without anything, let's start the class. Now, I would ask all of you one simple thing. I want every single one of you to name 3 different organisms. In the chat, go. All of you, quickly in the chat. Name 3 different organisms in the chat. I will say, let's say I will tell one fish, one tree, human. Tree, fish, human, or Homo sapiens. Any fish, let's take a shark as a fish. Let's take a mango plant, mango tree as a tree. 3 different organisms. Now I want everyone to tell me the same thing. Okay, same thing. 3 different organisms in the chat, go. All of you. 3 different organisms in the chat. Now students, all the organisms in this world, the large biodiversity, it could be a bacteria, all the way from a bacteria to a large blue whale or a fungus or a plant. Every single organism in this world is made up of same element. If I tell that line, will you believe me? Will you believe me if I tell you every single organism, me, you, plant, fish, bacteria, all of these organisms are made up of same type of chemical compounds? All of you will believe me, students? All of you will believe me? Yes or no? Tell me. All the living organisms are made up of same chemicals, right? All of us are made up of same chemicals. All of you believe me? You have to believe me, because I'm telling you right now. You have to believe me. But what are these elements? Students? The elements which I'm talking about is none other than the basic carbon, hydrogen, and oxygen, right? These are the basic elements or the chemical compounds present in every single living organism. You're like, yes sir, true. E. coli also is the same thing. But students, what if I tell you, let's take our earth. Let's take our earth right here. This is our earth right here. And some places we have water. We have water everywhere. Now students, can anyone tell me what do you call the innermost layer of earth? That's called as core. Then we have mantle, and then the outermost layer of earth is called as what, students? Outermost layer is called as the crust. Crust, mantle and core. Now students, if I take a piece of a crust, let's take a small piece here. Let's take it as Gokul's grandfather's property. Okay. From his property, I am taking one small piece of land. And when I analyze this non-living thing right here, I also find the same elements. I find the same carbon, same hydrogen, and same oxygen. The same carbon, hydrogen, oxygen is present in the grandfather property, and same carbon, hydrogen, oxygen is also present in living organisms. One is non-living and one is living. So if one is non-living, one is living, same elements, then what is the difference, students? What is the difference between both of them? Are they one and the same? The elements are same. Let me tell you, students, if you do the chemical analysis, the chemicals are same. The same elements are present. But the only difference in the non-living, when you compare it to living, is the relative abundance. Fancy term, no fancy term? I'll make it simple. The meaning of relative abundance is that the same elements are present in less quantity here. Less quantity. That's all. Less quantity. But the same elements from here, carbon, hydrogen, oxygen, is present in higher quantity in the case of living organisms. Same thing is present in a higher quantity. That is the only difference. Okay. Apart from here, we have cells. Here we do not have any cells. Okay. Now students, the cells remind me. The cell reminds me a cell. If you take a simple cell right here. Now students, if you take a cell, let's example this is a cell. Now students, can anyone tell me, if I do not have a cell wall, can I call it cell membrane or can I call it plant cell or animal cell? It does not have any cell wall, so it is an animal cell. Okay, let's take an animal cell. Now students, within this animal cell, I have a lot of molecules. Within this cell, I have a lot of molecules within this cell. And I will call whatever molecules present inside this cell part of a cell pool. I'll call it cell pool. Okay, cell pool. Now, within the cell pool, there are certain molecules which are present only in living cells. And the molecules which are present in living cells are called as what? Anyone? The cells or the molecules present in living organisms is called as biomolecules. So bio means living. Living molecules present inside a cell. Now within the cell pool, there is something called as micromolecules as well as macromolecules. So within a cell, we find something called as micromolecules as well as macromolecules. Both can be found within a cell. Now you might ask me, so what is the difference between a micromolecule and a macromolecule? Is it the size? Is it that micromolecules are smaller while the macromolecules are larger? Correct. But is that the only criteria? Is that the only way we can distinguish between micro and macro? Or is there any experimental proof to make us understand better the understanding of micro and macro? Is there an experiment? The answer is yes. So with that being said, with the basic framework, all of you on the same page, with the same basic framework, can we start today's class with the help of a simple, very simple experiment? Can we start, students? Can we start now? All of you? Okay. Can we start? All of you are understanding? All of you understanding what is living organisms? What are molecules present? Cell pool, micro, macro molecules. Yes, beautiful. All of you are very smart. All of you are very, very smart now. Now I want everyone to follow with me. I want you to write down a few things. And if you have any doubt anywhere, you can always ask me. So students, keep the energy going. If you know the answer to the question, keep answering. That's all I will ask all of you. Okay. Now, what is the experimental proof here? Now let's take a leaf here. Let's take a leaf. Let's take a leaf here. Or you can take a liver piece also, whatever you want. Or you can take a liver piece. Okay. Leaf or a liver piece. Liver piece, or you can take a leaf. Okay. And this liver piece and leaf, you need to add it. You need to add it into a chutney maker. I will call it chutney maker as mortar and pestle. Can you see this, students? This is the mortar and pestle. This is mortar and pestle. Mortar and pestle. Mortar and pestle. Now, once you add this leaf and chutney into this mortar and pestle, here, students, if you are making chutney, do you add water to the chutney? Do you add water to make it much more slurry, liquidy type? Yes, we add water. Water is basically a solvent. So, instead of water, we are adding some other solvent here. Solvent. The solvent which I am using here is called as trichloroacetic acid. It is called as trichloroacetic acid. Trichloroacetic acid. Trichloroacetic acid. Okay. So I will add trichloroacetic acid, that is basically Cl3COOCOH. Trichloroacetic acid I am adding into this mixture. Okay. Now let's use this color. Adding into this. Okay, done. Now I need to make a lot of nice, I have to mix it nicely. And after mixing nicely, we get a nice chutney here. Okay, this is the chutney which you get. I will call it as a slurry. Done? Done. Okay, I will call this slurry. Okay, done. Now students, let's come to this side now. Okay. Now you need to take a beaker. A small beaker. Okay. Now you need to take a small beaker like this. Take a small beaker. Okay. Now on top of this small beaker, you need to add a cloth, like a filter cloth, called as muslin cloth or cheesecloth, whatever you call it. You have to add it on the top, like a nice cloth is added, basically for filtration purposes. I'll call it muslin cloth. Okay, you can use a filter paper, you can use a cheesecloth, whatever you want. Okay, you can use it. Now, on top of it, we need to add a slurry. We need to add the slurry on top. Okay, done. Slurry has been added on top. Okay. Now, after some time, what happens? After some time, if you leave it for some time, because of gravity as well as the tiny pore size of the muslin cloth or the filter cloth, some amount of slurry will drop down. It will drop down. So, after some time, what happens is some amount of slurry is able to filter down or trickle down. Okay, some amount is trickling down. Okay, trickling down. Now, students, understand very carefully. This is the most crucial part. Okay. Now, here we have separate names for the amount of substance present at the bottom. Separate name for amount of substance present on the top. Okay. Some amount which is getting filtered down, this region is called as filtrate. I will call it as filtrate because it's coming down. Whatever is present on the top, student, this part here, whatever is present on the top is not filtered down, it is present on the top. I will call it as retentate. Now, the first difference is absolutely clear to all of you students. Yes or no? Tell me in the chat quickly. The first difference is clear to all of you. Whatever is present on the top is called as retentate, and whatever is on the bottom is called as filtrate. Filtrate, retentate. Okay. Now students, if something is falling down, technically it is soluble in the acid. Which acid? Trichloroacetic acid. Because it was soluble in the acid, it is able to filter down. So I will also call it this substance right here at the bottom as acid soluble fraction. Okay, acid soluble fraction. Now whatever is present on the top is technically insoluble in the acid. I will call it acid insoluble fraction. Okay. Now understand, all of you, understand. Now here what is present on the bottom, it is able to pass through the minute tiny gaps here. So students, you tell me in the chat, think logically. Do you think the size of the substances or the molecules present at the bottom, will it be large or will it be tiny? Or students, with the in respect to the size, usually we say the acid soluble fraction, the size of the organisms here is less than 1000 Daltons. At the same time, the size of the molecules present on the top is more than 1000 Daltons. It is going up to 10,000 also. 10,000 Daltons. It's very large molecules present at the top. Okay. Now students, you tell me, think logically and tell me again. If a molecule, the size is smaller, will I call it as micromolecule or will I call it as macromolecule? Think logically and tell me. So if the size is very less, can I call it as micromolecule? Okay. Micromolecule. What do I call here? I will call it as macromolecule. And every single substance present here are generally monomers. Tiny monomers can be present here. And here the substances are going to be polymers. Long chain polymers. So with that basic difference, you just did a simple experiment. With a simple experiment, all of you are able to tell me what is a micromolecule and what is a macromolecule. Now students, throughout this chapter, when we learn about carbohydrates, when we learn about nucleic acids, proteins, carbohydrates, anything, lipids, I will ask you the property of micro, I will ask you the property of macro. You should be able to tell me in the chat. If I ask you micromolecules, the size, what will be your answer? Tell me students, what will be your answers? What will be your answer? If I ask you the size of the filtrate or acid soluble or micromolecule, you should be able to tell me less than 1000 Daltons. Clear, all of you? Clear? This information should be fixed in your brain. Now students, this is not the end actually. This is not the end. This micromolecules and macromolecules can be taken and studied with different experiments later on, which is not given in a syllabus. This much is given, this much we have covered. Okay. Now, nice. Now students, a simple example, understand the example. Here I have given you, again table I have given you. All of these notes will be available in the Telegram channel. I am telling you now only. Before you later on tell me, sir, no notes you didn't give only, I'll give you, don't worry. Okay. Now, next thing is micromolecules example. Simple sugars. When I say simple sugars, I mean monosaccharides, disaccharides, yes, amino acids. It could be acidic, basic, neutral amino acids. Then we have nucleotides, single nucleotide. Now, if you look at the macromolecules, we have complex carbohydrates. Complex carbohydrates mainly include the polysaccharides. Then we have proteins. Obviously, Proteins are basically long chain of amino acids, so obviously the size is going to be larger. Then we have nucleic acids such as DNA and RNA. Now students, let me ask you one simple thing. Here under macromolecules, one kalla is there, or I will call it as imposter. Can anyone point out who is the imposter among the macromolecules? Anyone? Anyone? Who is the imposter here? Who is not supposed to be here but is hiding somewhere in the plain sight? Anyone? Who is the imposter? Lipids. Exactly. Exactly. This lipid right here, students, is very special case. This lipid right here is very special case. Okay. Why? Because students, the size of the lipid is around 800 Daltons. Around 800 Daltons is the size of lipid. So technically speaking, the size of the lipids is around 800 Daltons. Isn't it supposed to be in micromolecules then? Right? Yes, or makes sense. I should keep this in micromolecules. But no. Why? Because even though the size may be around 800 Daltons, this lipid is playing a game called as chain. This lipid right here is able to hold hands and make a structure called as vesicles. So it forms something called as vesicles, which I'll show you later on. Image is there, diagram is there. I will show you later on. Wait. But it forms something called as vesicles. And these vesicles are found in the retentate. So even though their size may be less than 1000 Daltons, but they are found in the retentate because of the chain or formation of vesicles. Is that clear to all of you? Students, is that clear to all of you? Yes. Now here is a simple comparison which I have told you in the starting. Earth's crust, human body, given your NCERT. If you notice here, hydrogen is higher, carbon is higher, oxygen is also higher. So the basic elements such as hydrogen, carbon, nitrogen is higher in the human body compared to Earth's crust. Simple thing which we learned. Okay. Now students, that was your first experimental proof for showing all the organic compounds. Now, a human body. Do you think we also have inorganic compounds? Do we have certain minerals in our body? Yes or no? Tell me in the chat, that's all. Just tell me yes or no. Do we have certain minerals in our body? Like certain inorganic substance in our body? Yes or no? Tell me. All of you. Do we have them? Do we have them in our body? You're like, yes sir, obviously. We cannot survive without minerals because minerals are cofactors. ATP synthesis cannot happen. Yes or no? Yes. We cannot do muscle contraction without the calcium. All of you know that muscle contraction, myosin contraction. So minerals are extremely important in our body. So how do we analyze them? There is an experiment again. Now students, are you ready for the second experiment now? Ready for second experiment? All of you? Okay. Obviously you're ready. You're my students. Now again students, we need to take a leaf again. Okay. We need to take a leaf again. Take a leaf again. Okay. This time we are not doing a chutney directly. Okay. This right here is a fresh leaf. Fresh leaf. But this time what are we doing here is, this fresh leaf, we are doing a process called as removing the water. What do you call it, students? What do you call the process of removing of water? It starts with the E. What do you call that? What do you call it, students? All of you? Evaporation. Okay. Now once evaporation is done, okay, you will have one more leaf. All the water content is gone now. Okay. Now if this was called as fresh weight, this is called as a dry weight, because all the water molecules are been removed right now. They have been completely removed. Now what do? Next step is grinding. Okay, next step is completely grind it. Grinding can be done in mixie only. Mixie. You know, all of you have mixie in the house, no? Grind it nicely. Then after grinding it, okay, once you get the powder, you need to do something called as incineration, or simple word is burn it nicely. Burn the powder. Okay, burn the powder. Now, once you burn the powder, what do you get, students? We get something called as ash. Not the Pokemon ash, normal ash. Okay. Once the ash is obtained, what exactly is happening here, students? Oxidation is happening here. Combustion, basically combustion. When oxidation is happening, all the carbon compounds are burned. Yes. Remaining water also has been removed. It has been removed. Everything is removed. So we end up getting the ash. Now, students, if you take this ash right here and you do analysis, right? If you analyze this ash, we will obtain all the information regarding all the inorganic substances, because this ash does not have any organic substance, it does not have any water molecules, it is only left with inorganic substances, and we can do further analysis to obtain a desired result. Okay, and this entire experiment is called as ash test. And when you do this experiment, this is what you get: a list of representative inorganic constituents of living tissue. Sodium, potassium, calcium, magnesium, and compounds. Everything is present in the inorganic way. Okay. So first one is organic, where we made a slurry, and second is inorganic test. So with these two basic tests, we know right now a human body or any living organism has certain type of organic compounds and certain types of inorganic compounds. That was our basic understanding or derivation from these two experiments. Is that clear to all of you? Students? Basic things. Okay. Now, if that is clear to all of you, simple ash test, can we go to the next concept called as amino acids? Okay. Now, I'll ask you all of you a simple basic question. The basic question is that, students, all of these amino acids, right, we keep hearing them. What will happen if I take one amino acid, one more amino acid, and one more amino acid, and if I completely join them, what do I get? Can anyone tell me in the chat? What do I get, students? If I join all the amino acids, what do I get? All of you. So if I'm joining amino acid, can I call amino acid as a monomer here? Amino acid is a monomer. Now if I join multiple amino acids, I will end up getting the protein. Here. Okay. Now question to all of you. Okay. This amino acids, this random amino acids or this protein, right? Where do we get it from? Right? From where do we obtain all of these different types of amino acids? Do we buy them in the shop? Can we buy 1 kg amino acids? Is it possible to buy 1 kg amino acid or 1 liter of amino acid? Is it possible to buy? Or do we get amino acids from some other form? Or do we make this amino acids on our own in our body? Can we produce them on our own? All of these questions will be answered in the next 5 minutes. Okay, next 5 minutes I can explain all this. Now, understand this. We learned all them are joined together. They make up the protein. Now, students, there are millions of proteins, and millions of proteins are there. All of you might be aware of trypsin, melanin, actin, glucagon, rhodopsin, collagen, creatine. All of these are different types of proteins. Now, you might be wondering, if there are millions of proteins, how many amino acids might be there? Try to imagine. Millions of proteins are there, different types. Obviously, there should be at least around 100 amino acids, 200 amino acids. How much do you think there are, students? How much do you think there are? To make up this entire millions of proteins, we must have so many amino acids, right? So many amino acids. But the reality is what you know? There are just 20 amino acids. What? Yes. To make up the millions of proteins in the world. Millions of proteins in the world. All the way from the proteins in our body to the snake, plant, bacteria, everything. There are just how many students? 20 amino acids. Isn't life amazing? Now you tell me. Isn't life amazing? Just 20 amino acids is making up millions of proteins. How fascinatingly complex this arrangement should be, right? 20 amino acids. Just 20 amino acids are there. Okay. And all of these amino acids, students, some amino acids are present inside our body. Some amino acids are present, we take from outside. Like all of you love, right? Chicken, mutton, pulses, right? Avocados and everything. Avocado has mostly fat and everything, but yeah. So all of these amino acids can be taken from outside, but also produced in our body. Okay, remember that. Now, based on this mode of intake or mode of requirement, amount of requirement, there is a classification. It's called as essential amino acids and non-essential amino acids. Can anyone tell me the difference now? Anyone? What is the difference between essential and non-essential? See, essential means it is important to us. It is important. Both are important actually. Both are important. But the difference arises in that even though it is important, our body, students, unfortunately, body does not produce. Our body does not produce this type of amino acids, essential amino acids, does not produce it all. So if the body is not producing this, how do we obtain this then? How do we obtain this? It is obtained through diet. The foods we take, right? The fish, chicken, everything. So essential amino acids are derived from the diet. Now what about non-essential amino acids? Non-essential amino acids are produced by the body. They are produced by the body. All of you understood? All of you understood? So if the next time, all of you might be wondering, do you remember this? Something called as a protein powder. How many of you know? All the people who go to gym, right, including me also, all the people who go to gym, right, they take something called as protein powder. Now students, there is a big scam here. What is a big scam? Before you buy any protein powder, always look at the label behind. Why? Because the protein powder, yes, they'll give protein powder. But what if, what if all the protein which they're giving you, what if it has non-essential amino acids? Scam no then, all of you tell me, scam no then? Big scam, right? They're giving you protein. They're giving you protein which is already produced by the body. What is the use then? What is the use? Our body is already producing it. So always be careful before falling into the scam there. Okay, before falling into the scam, I'm telling you now only. Always look for amino acids which are not produced by the body. That is, essential amino acids are present in the whey protein or protein powder which you take. Okay, small, right, small thing. Now, I told you so much. I told you about essential, non-essential. Now, what are these essential? Out of these 20 amino acids, which are essential, which are non-essential? I will also tell that. Listen to me now. Listen to me. See here. All of these are essential amino acids. All of these are non-essential amino acids. Right? Non-essential amino acids. Now, next time when you're buying protein, right, always look for all of these amino acids, especially students, especially lysine, leucine and valine, right? These three amino acids has to be there. Okay, has to be there. So please be careful before buying this protein powder. And also students, always try to look for these amino acids in your diet also. Yes, dal, all of these has protein, but always make sure that you have essential amino acids getting into your body. Okay. Is there a small trick? This is a small trick, students. Private Tim Hall. Phenylalanine, valine, threonine. Tryptophan, isoleucine, methionine. Histidine, arginine, lysine, leucine. All of these are essential amino acids. Here, almost all guys get cut after going to play sports All of these are non-essential amino acids. A small trick for you, download it, use it. Okay. Now, what do I say? Students, I'll tell you one from personal experience, small story. Listen to me very carefully. Small story. I was one of those kids who used to rely on tricks. See, I love tricks still, I love tricks. But let me tell you what happened to me once. Okay, after I learned my lesson, and I do not want any one of you to do the same mistake which I did. Okay, honest mistake. So I learned a small trick and I went to the exam hall. I learned around 4 or 5 different tricks for almost the same concept. Okay, a small concept. But when I sat in the examination, the trick became tricky. I could not remember the trick only because the trick was so tricky that I did not practice it enough and I went wrong and I lost the marks. Simple example I'll give you. Here also we have T, that is tyrosine. And here also, students, we have T for tryptophan. So here also we have T, here also we have T. But if you don't practice it, if you don't practice this T difference, where tryptophan comes and where do we get tyrosine, I am very sure all of this trick is meaningless. You will still lose the marks just like me. So do not make the same mistakes as I did. Learn from the mistakes of your elders. Okay, learn the mistakes from your elders. Do not repeat the same mistake. If you're using a trick in examination, practice the trick beforehand and then go to the examination. Don't sit like one duck there later on. Okay. Oh God. And now, students, with that, we come to the structure of amino acid now. Structure of amino acid. This is where many students find it difficult to understand, but I'm going to make it easy for you. Ready, all of you? Ready? Ready? Okay. Now, students, understand, all of you. Tell me, what is the chemical formula of methane? All of you. What is the chemical formula of methane? Tell me in the chat quickly. Quickly, all of you in the chat. Okay. Yeah. Oh, trick becomes tricky. Yeah. All of you drink some water also now. All of you drink some water. Done? Okay. Tell me. CH4. Absolutely correct, all of you. Ravindrani, Daisy, all of you correct. It is CH4. Right, CH4. Let's draw it here. Let's draw it here, students. C. Done. All of you wrote. In the books, I want everyone to write down now. Now, what is an amino acid? If you open your NCERT right now, there is a line there. Amino acids are substituted methane. Okay, so we take a methane right here and we need to play a small game here. What is a small game? I will give you H, you are supposed to give me one functional group. Ready? Okay. I will give you this hydrogen. Students, I will give you this hydrogen. You will give me something called as carboxylic acid group. You will give me something called as carboxylic acid group. Carboxylic acid. Okay, done. Now again, I will give you this hydrogen here. You are supposed to give me, students, what will you give me? NH2. What is this? This is an amino group. This is a amino group. Okay, done. Now, students, now I will give you this hydrogen again. You are supposed to give me some type of functional group. You're supposed to give me something called as a functional group. Done. So the basic structure, students, what is the basic structure of a amino acid? A carbon with a carboxylic group on one side, amino group on side, hydrogen on the other side, and a functional group on the side. This right here is a basic structure of a amino acid. Okay. Now, students, we can replace this R. All of you, we can replace this R functional group with any kind of functional group and we get amino acid. I'll show you a simple example, understand. Simple example. Okay. Let's write the group here one more time. So what do we have, students? We have a carbon in the center. Then we have carboxylic acid, amino group, and we have a hydrogen, and we have a R. Okay, and we have a R. Now students, what if this hydrogen right here, so this R here is a hydrogen. If this is a hydrogen, what do we get, students? If this R is a hydrogen, we end up getting an amino acid called as glycine. Called as glycine. Glycine right here. A few things about glycine, students. A few things about glycine right here. Remember, students, glycine is a simplest amino acid. Simplest amino acid. Yes, simplest amino acid. This is the only amino acid which is non-chiral in nature. Non-chiral in nature. What is the meaning of non-chiral? Imagine if this carbon, which is the alpha carbon right here, if it has all four neighbors as a different element, then it is a chiral carbon. Okay, different chiral carbon. But right now, in the case of glycine, hydrogen here. So it is not different. All four is not different. So it is going to be non-chiral nature. Non-chiral nature. Also, it is optically inactive. Okay, it is optically inactive also. Inactive. Optically inactive. Okay, it is the only non-chiral amino acid. Glycine is the only non-chiral amino acid. It has all four is not different. Here we have hydrogen, here also we have hydrogen. Okay, done. Next point you understand. Now what if this R group is said to be a methyl group, students? Methyl group. What if it is a methyl group? Then what do we get, students? If this functional group, this R is methyl, then we end up getting an amino acid which is called as alanine. Called as alanine. Alanine. Okay. Now, next last one. What if this R group is said to be a hydroxymethyl? Hydroxymethyl. Then what do we get, students? If this is a hydroxymethyl, then we end up getting a amino acid called as serine. Called as serine. Okay, serine. So what do we get, students? First one was glycine. If it is H. If it is methyl, it's alanine. If it is hydroxymethyl, CH2OH, then it is a serine. These are the three different amino acids which structure is given in your NCERT and you have to remember at any cost. Will you remember? All of you wrote down? All of you wrote down? Done? Tell me in the chat, students. And all of you, the 100 likes has not been done yet. So quickly smash the like button. The target is 200 for today. 200 likes is the target. Let's make it happen. Okay. Now, if this is done, students, I want everyone to remember this. Write down this. Once you are done remembering this, tell me done sir in the chat, fire emoji in the chat, and then I'll go forward. Because I want you to understand this now only. Do not keep it as a backlog. We are trying to clear backlog today. Done? All of you. Try to remember this also. So if it is hydrogen, glycine. If it is hydroxymethyl, it is serine. If it is methyl, it is alanine. Done, all of you? Noted, all of you? Yes? Exactly. Gasser. What gas? Okay, gas. See, okay, gas. G for glycine, A for alanine, S for serine. Okay, remember gas, students. Remember gas. This doesn't need a trick. This doesn't need a trick, but if you want, you can keep it. Okay. Now students, how many of you saw my recent fact? How many of you saw my recent fact on the channel? How many of you saw? Recent fact on the channel, something about skeleton flower. So students, if you want more such facts, right, because I have a lot of botany facts and all of you are aware. I have almost around, I have a fact book where I have written more than 30 to 40 facts right now written. All of them can be made into a separate video. So if you want more botany facts from my side to you, let me know in the comment section of that video. Okay. Now, like for example, I can tell you about a fungus which can control mind. Yes. I can tell you about a plant called as walking palm tree, which can actually walk. I can tell you about how plants can talk to each other. They can talk to each other. Right. I can tell you about a dragon wood tree. It is called as red dragon wood tree. If you cut the tree, you get blood out of it. It is not blood, but it is the red color sap. So it is bleeding red. Okay. So there are so many facts. I'll tell you someday, one by one, botany facts. Okay. Next thing is, students, sulfur containing. There is something called as methionine and cysteine. Now this reminds me, students, methionine. Did you study this in the chapter called as molecular base of inheritance? What does methionine there did we learn? Something called as start codon. How many of you remember? How many of you remember, students? Something called as start codon. How many of you remember that? Raise your hands. So these are the sulfur containing different amino acids. Let me tell you now only. Let me tell you something. Here you do not need to remember the structure. No. Remember the terms. Remember the names. So methionine as well as cysteine are the two different types of sulfur containing amino acids. Please remember that. Once that memory is fixed to your mind, just say done sir in the chat. So methionine as well as cysteine, two different sulfur containing amino acids. Now students, there is one more classification. Classification of amino acids based on the structure. What do I mean by that? Okay, there are two broad classification here. The first one is called as aromatic. Now quickly, how many of you know what is aromatic? Aromatic means amino acids which have, students, amino acids which have a ring. They have a aromatic ring in the structure. Like they have a benzene ring. Basically they have a benzene ring. All of you know what is a benzene ring, right? All of you know what is a benzene ring? So these amino acids have a benzene ring in their structure. Okay, benzene in the structure. Okay. Now what about other one? They are called as non-aromatic, or we can also call them aliphatic. Non-aromatic, or we can also call them aliphatic. Chemistry, you know so much, students, chemistry. How many of you like chemistry, students, more than bio? Here we have straight chains. I love chemistry. Chemistry is fun. Okay. So they have straight chain here. Long chain of amino acids. Simple difference. I hope all of you know this. Now students, here are a few examples of aromatic amino acids. Again, structure is not important. Names are important. Now students, I'll help you to remember it. Okay. Phenylalanine. How do you remember? How many of you remember something called as PKU disease? Students. How many of you remember PKU disease? Phenylketonuria disease. Which chapter? Tell me. Which chapter? Quickly tell me in the chat. Students, smash the like button and tell me the answer, all of you. Which chapter? PKU disease, phenylketonuria disease. PKU disease, we learn it in Principles of Inheritance and Variation. PKU disease is a autosomal recessive disease, right? PKU disease is autosomal. All of you know that. I don't need to write. Okay. In this disease, what happens is phenylalanine is not converted to tyrosine. So all of you can remember now. So next time if someone tells you aromatic amino acid example, remember which disease? All of you should remember PKU disease. In PKU disease, what is happening? Phenylalanine is not converted to tyrosine. Okay, so two examples done. Okay, two examples done. Now, how do you remember this? Tryptophan. Tryptophan is used in auxin preparation. Used in auxin preparation. Now, which chapter? Tell me. Which chapter again? Which chapter? Auxin preparation. Where did you study? In the plant growth regulators. In your plant growth and development, we study that. So, all of you can remember. So, this is how you can remember, students. There is always not a hard and fast rule that you need to by heart everything. You can remember things by connecting them. Like how you can connect aromatic amino acids to PKU disease, learn these two. You can also learn tryptophan by auxin preparation. Done. Okay. Now, last type of classification based on the functional groups. There is something called as acidic amino acid, basic amino acid, neutral amino acids. Very, very simple. Very, very simple. In fact, I will teach you right now. Listen to me very carefully. Very simple it is. Very simple. Okay. Now, all of you look at this diagram where we have two examples of acidic amino acids. Okay. If you look at this diagram, how is it different from the normal structure of amino acid? Tell me in the chat. These two diagrams, these two structures, not diagrams, these two structures and a normal structure. What is the difference between them? Tell me. What is the difference? Tell me quickly in the chat, all of you. What is the difference? Look at the structure and tell me. What is the difference? See, I want everyone to use your brains little. Use the brains and tell me. What is the difference between these two structures and a normal structure of amino acids? Think, just because if I tell you it's no use. You should try on your own little bit. Hard work. Exactly, all of you are absolutely correct. See, here we have a carbon, one carboxylic acid this side, amino group this side. So this hydrogen is supposed to be on top. Okay, I can write here. Now this is the R group. This entire thing is the R group. Okay. Now do you notice in the R group here, do we have an extra? Do we have an extra COOH group here? An extra carboxylic acid group here? Same goes for your glutamic acid also. This is the amino group, carboxylic, hydrogen. This entire thing is the R group. R. Now here do you notice again? Do you notice here again, students? There is an additional COOH, that is an extra carboxylic acid group. Extra carboxylic acid group here, COOH. So that is the reason. That is the reason they are called as aspartic acid and glutamic acid. Tell me, students, how many of you understood the concept right now? See, I can make you by heart this. There is a very easy way to by heart it, but my students should understand why it is called as, acid in nature. Now, again, I will show you the diagram structure for basic amino acid. Tell me how many of you can find the difference in it. Okay. Tell me, students. What do you find difference again here? In basic amino acids, which we have lysine, arginine, and histidine, what is the extra thing here, students? Tell me. All of you tell me. What is the extra thing here? Just think logically and tell me. Let me drink some water. I'm feeling very thirsty today. Extra? What do we have, students, here? Extra amino group we have here. Yes. Do you have extra amino group here? So all of these are called as basic amino acids because they have an extra amino group. Now students, how many of you remember? How many of you remember lysine and arginine? These two examples from the chapter molecular basis. Where do we study this, students? Lysine, arginine. In which statement do we study? Does it have anything to do with something being negative charged? Basic in nature? Anyone? Where do you study this? Where do you study about lysine, arginine? In the chat, molecular basis. Now, neutral amino acids. Nothing. Neutral. Everything is neutral here. Nothing. No extra COH. No extra NH2. Nothing. The example given your NCERT is valine, but I have given your glycine here, not valine. Okay. Now, students, with that, we come to the end of amino acids. Okay. So, what we say, students? We said that there are totally how many amino acids? Tell me. There are totally 20 amino acids. Oh, why did this come here? Okay. There are totally how many amino acids? Total there are 20 amino acids. In the 20 amino acids, we have 2 are acidic in nature, 3 are basic in nature, and remaining 15 are neutral in nature. Okay, aspartic acid, glutamic acid, lysine, arginine, histidine, remaining all are neutral amino acids. Okay, now students, how many of you find it difficult to understand the concept of Zwitterion? Raise your hands right now, because I'm going to make it easy for you in the next 5 minutes. In the next 5 minutes, I'm going to teach you Zwitterion concept, and this will become easiest concept of your life. Tell me, how many find it difficult? How many of you find it difficult to understand the concept of Zwitterion? Zwitterion. Tell me in the chat quickly, all of you. Be honest, don't lie. No point in lying with me. Okay, I will not get anything, you will not get anything. Okay, if you don't know, learn it today. Okay, Zwitterion. Okay. Now, students, have you noticed that? You are sometimes different with different people. Okay, I'll give you a simple example. Okay, nicely you're playing games in the house. Suddenly dad is entering the house. What do? Switch off the computer, switch off the laptop. One nama you'll put like, Papa, come inside. Do you want water? Where are my books? Books are hiding. You'll keep open your books. All the books, all possible books. Classwork, homework, NCERT, and with Tattva books, everything will open. Everything will open and keep off in the book. And as if you're studying from morning, you'll act. Okay. So depending upon the situation, did you change? Yes, you changed. So your amino acids are just like you, students. Amino acids are just like you. They are not very different from you. Okay. Like how you will react. Okay, before the belt is coming out. Like how will you react, right? Same way amino acids also react. Amino acids will change themselves depending upon the media around them. Yes, they will change. Okay. So different pH, different state of amino acid. So in acidic pH, amino acids are acting different. In basic pH, amino acids are acting different. So depending upon the pH, the amino acid structure changes. With that basic information, I will show you some diagram. I'll show you structure, learn it. Okay. For example, let's take an amino acid. Okay. Now, this is a random amino acid. This is a random amino acid, and I'm keeping it in acidic medium. In acidic medium, what do we have, students? In acidic medium, we have many H plus. Lot of H plus are there. So many H plus. Now these H plus is taken up by amino group. Okay. So in acidic medium, what happens? In acidic medium, your amino acid is getting converted to actually basic in nature. this is going to become NH3. Okay, this is going to be NH3. So in acidic medium, it is becoming basic in nature. Now similarly, let's take one more amino acid. Let's take one more amino acid. Now I'm going to keep it in basic medium. What will happen? If I keep it in basic medium. In basic medium, what happens? This hydrogen is lost. Hydrogen lost. So in the case of amino acid in basic medium, it is becoming acidic in nature. Tragedy. Okay, so depending upon the medium, right? Depending upon the medium, the amino acids change. Okay, what do I mean? Experience that still happens in the house. What do you think? I can keep playing games all day. Not possible. I can keep working all day. They don't have a problem. Okay, so what do you mean? It's not experience. It still happens in the house. Okay, don't worry. Same. All of us are in the same boat. All of us are in the same boat. So I have to pretend sometimes. You know, I'm studying always. I'm working always. Okay. The only difference is you're showing studying. I'm showing working now. So I hope all of you understand the simple basic concept that in acidic medium, amino acid is becoming basic. In basic medium, it is becoming acidic, right? So amino acids are changing based on the location or based on the media present around it. Okay, based on media around it. Simple as it is. Okay. Now, how does that relate to your Zwitterion? How does that relate to Zwitterion right now? And how do we understand that concept here? Very simple. Very simple. Just follow through. Okay. So what happens here is understand. Okay. Now, all of you please notice this one. This is the main thing here. See, this one we discussed, right? In here NH3 plus. This happens where this happens in the case of acidic medium. Acidic medium we get this. We get this CO minus in the case of basic medium. Yes, basic medium we get that. Okay, we get that in basic medium. Okay, now understand. Okay, understand all of you. Let me tell you one more. This is the carbon, carboxylic acid. Here we have your amino group NH2. Here we have your hydrogen, and here we have R group. So understand what happens here, all of you. Listen to me very carefully. This hydrogen is sent this side. This hydrogen here is transported to this side. What do you call that? This is called as, at a particular pH, what happens? Intramolecular proton transfer. Intramolecular proton transfer. It's called as intramolecular proton transfer. So this molecule, this proton is being transported this side, and this entire thing, when it happens, we end up getting a structure like this. Can you see? COO minus, this hydrogen is gone, and here we have NH3 plus. So positive charge here, negative charge here. So at a particular pH, at a particular pH, we get a molecule. Which has positive charge on one side, negative on one side. This molecule right here is called as Zwitter form. It is called as Zwitter form. Okay. And when I'm talking about at a particular pH, right, this particular pH is called as isoelectric pH. Isoelectric pH. So at isoelectric pH, what is happening, students? At isoelectric pH, intramolecular proton transfer is happening, and we end up getting a molecule which is either positive charge or negative charge at the same time, where the net charge is 0, and such molecule is called as, such ion is called as Zwitterion. It's called as Zwitterion. Here, a molecule, one functional group is having positive charge and other having negative charge. Positive is equal to negative, net charge is 0, and this entire thing happens at isoelectric pH. Done. How many of you understood? Raise your hands quickly, all of you. How many of you understood? Raise your hands quickly, all of you. Quickly, raise your hands. Isoelectric pH. All of this happens at isoelectric pH. Done? Done. One question here. Tell the answer in the chat quickly. Tell the answer in the chat. Identify the basic amino acid. Identify the basic amino acid. And all of you like that video quickly. All of you like that video. Not video, this video only. 200 is the comments. 200 is the target. Answer in the chat quickly. Identify the basic amino acid from the following. Very easy question. I'll give you 10 seconds. Done? This question again. NEET 2024 question. The following can form a Zwitterion: a fatty acid, a monosaccharide, amino acid, nucleic acid. Quickly answer in the chat, all of you. Which can form a Zwitterion? NEET 2024 question, all students who wrote last year. Exactly, the amino acid. Amino acid came from Zwitterion. Okay. Now students, before we start lipids, right, before we start lipids, I wanted to talk to all of you a little bit, okay. Students, your exam is very near, all of you are aware, right? All of you are aware. We have done the entire class 12th to some extent, right? The bio club. Now we are doing backlog series because I am taking high weightage chapters of your class 11th. Gopika ma'am is doing the same to clear all your backlogs, right? So with this much preparation, I want everyone to tell me, out of 10, what is your preparation at this point? Rate your preparation out of 10. Tell me, students. Rate your preparation out of 10 right now. Right now. Let me see in the chat. Because all my students are working really hard. We are working really hard here. Right. So I want to see how much is your preparation coming along, how much hard work you are doing, and how much effort you're putting in. What is still requirement? Right. Do you need to solve more questions? It's coming up. Gingerly oil is coming up, I know. Okay. So tell me what is your preparation stance right now, because it's very important right now, because I want everyone to be on point, solve the questions every single day, 200, 300 questions, keep solving. 8, 4, 9, very good, very good, very good all of you. Now I'm telling you students, let us know in the comment section what can we do from our side to help you more, because all of you are aware, right? We as a team here, we're always here for you. Like, we have been working hard for a very long time, right? So we want every one of you to get a really good medical college, not just any random medical college, not just any random medical college. We want you to get into good medical colleges, because that is where your future is going to be, right? So promise me, all of you, you'll put extra hard work from today onwards, extra question solving from today onwards, and make the dream into reality, right? Make the dream into reality. Okay, so we'll make it happen. More question solving, we'll make it happen. Don't worry, we'll make it happen. So PYQ sessions, we will do it. Okay, so all the best. Let's get back to the class now. Okay, small talk. I wanted to talk to all of you because, right, it's been a due that I spoke to all of you a little, right? So practice session will happen. Don't worry. Okay, keep confidence. Keep solving questions every single day. From our side also, we'll give you more. Right, from our side, we will give you more and more content, right? If you want, I will try uploading more PYQs, right? So that is why your future is going to be bright. Okay, so all the best. Let's get back to the class now. Okay, it's small talk. I wanted to talk to all of you because, right, it's been a due that I spoke to all of you a little, right? So if all of you want, if all of you can solve, if you want to solve, today I will sit and make around, let's say, I will give you DPP questions. I know this takes a lot of effort. This takes a lot of efforts. So if you are up for it, if you're up for it, today's class, after today's class, I'll give you DPPs also in a Telegram. I'll make it happen. So just that I'm a little lazy also, like all of you. Okay, I'm gonna have to sit and make the questions. Questions are not randomly available from everywhere, right? I'll also make it happen. Okay, students, let's go. Now with the same energy, and the same Josh, can we start the second part of the chapter, next part of the chapter, that is called as lipids. Can we start? Now I believe till now, students, all of you do not have any doubt whatsoever. We finished lipids, we finished proteins to some extent, we finished amino acids to some extent. Now we go to lipids now. Can we start lipids now? All of you. Can we start, all of you? Yes. Do not let the energy go down, because I want everyone to be very active today, because it's going to be a very long lecture. This is a very big chapter, all of you know. This is a very big chapter. It takes a lot of dedication to sit through this entire chapter. Right? So show that. Now, how many of you tried this? Raise your hands. How many of you tried this? Raise your hands. Have you ever tried mixing oil plus water? If you have tried, does it mix? Tell me in the chat. If you have tried mixing oil plus water, does it mix at any cost? Have you tried? All of you? Have you tried mixing oil plus water? If you have tried, you might have noticed initially it does not mix. Initially it does not mix. But if you keep shaking it, for example, if you take a bottle like this and if you keep shaking, at one point it will mix. At one point, oil and water, both of them will mix, and that is called as emulsion. Okay, that is called as emulsion. But technically, students, technically speaking, lipids do not mix with water. They do not mix with water. Right? Lipids weigh less than 800 daltons, but still they are present as acid insoluble fraction on the top. Why, students? Answer is here. That is, lipids, right? They play a game called as chain. I told you already, chain game. All of you have played, right? All of you play chain game. You hold hands. Some will say chain, cut. Same thing they are doing. So when they play this entire chain game, they form something called as vesicles. And these vesicles are present on the top. They are insoluble in the acid. Okay, they are called as the vesicles. Now, lipid students, tell me, are they present in acid insoluble or acid soluble fraction? Tell me in the chat. Where do you find lipids? Acid insoluble. They are found in retentate or filtrate. They are found in retentate. What is the size? Less than 800 daltons. Okay, 8 needles. Okay, they are technically insoluble in water unless we force. Okay, they are not polymeric. Not polymeric in nature. Now, lipids, there are under simple lipids, which is given your NCERT. Under simple lipids, there are two main types. Okay, one is called as fatty acids, and the other one is called as glycerol. These two are different types of simple lipids. But do we also have other type of lipids? Is it given in your NCERT? Yes, it is given. But they don't tell it in the statements. They tell you in the diagram also mainly. Let me show you what are those. Okay. Now, there is something called as simple lipids, which we just now learnt, where we have fatty acid and glycerol. Then there is something called as compound lipids. Example I'll tell you now only, I'll explain later on. The example for compound lipids given in your NCERT is lecithin. We'll come back to that. Derived lipid is also given in your NCERT. That is cholesterol. Cholesterol is a derived lipid. Okay. So all of this is given in your NCERT, but in a very here and there, they've thrown here and there, not properly made into a segments, but I'm going to teach you properly here. Okay, so simple lipid, compound lipid and derived lipid. Okay, first we'll focus on simple lipids. Simple lipids. Now under simple lipids, I told you there are two types, fatty acid, and students, tell me in the chat quickly, all of you, under simple lipid what do we have? Under simple lipid we have the fatty acids as well as glycerol. Now one by one I want you to understand what is fatty acid and what is glycerol. Very simple. Understand the structure first of all. All of you understand the structure, it is very simple. Okay. First you need to understand what is fatty acid. Understand what is fatty acid first of all. Okay. Fatty acid is nothing but carboxylic acid plus R group. It is nothing but carboxylic acid plus the R group. What do I mean by that? Understand here. Understand the structure. Let's take, we have a R group here. R group. To this R group there is a carboxylic acid. That's all. This right here is called as a fatty acid. This right here is a fatty acid. Here we have the carboxylic acid group, this part here, and here we have a R group. This is the functional group. Functional. Now the interesting part here is this R group, right, this functional group, this can be methyl, this can be ethyl. All of you know what is ethyl, right? C2H5. Or this R group can be between 1 to 19 any CH2 groups. Long chain of CH2, 1 to 19 carbons, long chain of 19 carbons. Everything is possible, this R group can be. Okay, so R group can be any of these, and this is the carboxylic acid. Together this entire thing is called as a fatty acid. Fatty acid. Why acid? Carboxylic acid. Fatty because of the entire R groups here. So fatty acid group. Right? This is the entire hydrocarbon chain, carboxylic acid. Just so simple it is. Nothing complicated. So simple it is. Okay. Now, your fatty acids can be broadly classified into two types: saturated and unsaturated. Simple difference. Write down, all of you. Write down. The first and the most important difference is the bond. In the case of saturated fatty acids, it's mainly single bond. It is mainly single bond here. Single bond. Now this one, unsaturated, they will have double bond between them. They will have double bond between them. They will have double or even triple bond sometimes. Triple bond. Double bond, triple bond. Okay. That is the basic difference. Now, saturated fatty acids, students, generally, I'll give you an example, then we'll learn. Okay. The example for saturated is butter and ghee. Okay, butter and ghee. Now tell me, students, this butter and ghee, right? Does it melt very easily compared to oil? Tell me. Does it become hot very fast compared to oil? No. Oil, cooking oil become very fast. Oil melting, they'll become boiling point, melting point is very high. But ghee or butter a little difficult, right? Because they also have very high melting point actually. Okay, right? But compared to oil, your saturated fatty acids, the melting point is much more higher here. And ghee or butter, they're solid at room temperature. You might have seen ghee and butter is solid at room temperature. They are butter at room temperature. Okay, that is saturated fatty acids. Unsaturated fatty acids, melting point is low and they are liquid at room temperature. Liquid at room temperature. Okay. Example is cooking oil. Cooking oil. Now here your NCERT tells me one more example called as gingerly oil. Gingerly oil. Who names gingerly oil, man? Seriously. Gingerly oil. Example here. Okay. Now apart from that, here also we have two other examples under saturated. The two examples given in NCERT is palmitic acid and stearic acid. Palmitic acid is 16 carbon and stearic acid is 18 carbon. Okay, 18 carbon. That is under saturated. Okay, unsaturated acid. Then under unsaturated fatty acids, there is a 20 carbon compound here. Here we have something called as arachidonic acid. This is around 20 carbon. Clear all of you? Clear? Tell me, students. It's not ginger. I don't know, students. I think I have to do some research now. I think I have to do some research on gingerly oil. I think it doesn't have any ginger or per se. I don't think it has some ginger or anything. Okay. So here we have saturated, unsaturated, single bond, double bond, high melting point, low melting point, solid room temperature, liquid temperature, butter, ghee, cooking oil, palmitic acid, stearic acid, arachidonic acid, right? All of these are examples and properties of saturated and unsaturated fatty acids. Okay. Now students, apart from that, there is a small concept called as MUFA and PUFA, right? Under unsaturated fatty acids, there are two types here actually. Something called as MUFA and PUFA. What is the difference? Understand. Okay. Now if you notice, all of these are 18 carbon. This is 20 carbon. Okay, 20 carbon compound. Now what is MUFA? MUFA is a unsaturated fatty acids. Okay, with a single double bond. Single double bond. So they will have only a single double bond. That is called as MUFA. Can you see here? Oic acid is an example of MUFA. It has a single double bond, only one double bond. In this entire chain, there is a single double bond that is called as MUFA. Then what is PUFA? PUFA is polyunsaturated fatty acids. All of these, 2 double bonds, 3 double bonds, 4 double bonds, all of these are MUFA. Okay, so we have PUFA and MUFA. PUFA has only a single double bond. PUFA will have multiple double bonds. Okay. Now students, tell me, how many of you are very rich? How many of you use olive oil in the house? How many of you use olive oil in the house? Tell me in the chat quickly. Raise your hands. Full flexing on all of us. In my house, we use sunflower. Tell me, how many of you use olive oil in the house? They say, right, students, they say olive oil is good. It's very good. It's very healthy, right? It's very healthy. Olive oil, right? Why? Why do they say olive oil is healthy, students? The answer is very simple. First of all, it's an oil. So if it is a cooking oil, if it is generally oil, tell me, is it going to be saturated fatty acids or is it going to be unsaturated fatty acids? This is going to be unsaturated fatty acids. But this unsaturated fatty acids is a MUFA. Okay. It's going to be a MUFA. This one, MUFA. And it also has something called as omega fatty acid. It has something called as omega fatty acid. Okay. Now the best thing about olive oil is that this olive oil will reduce the bad cholesterol. It will reduce the LDL. What is LDL, students? Low density lipovolume, right? It will reduce the bad cholesterol in the body. Bad cholesterol can cause the heart attacks and everything, right? So olive oil will reduce the bad cholesterol in our body. Okay, it's very healthy. Now, anyone, any doubt in fatty acids? Saturated, unsaturated? Structure is here, fatty acid. Now we come to something called as glycerol. Glycerol is even more simple, students. Glycerol is nothing but an alcohol. It is basically an alcohol, that's all. You learn the structure, everything is over. Learn the structure. Here we have 1, 2, 3. 3 carbon is there. 1 OH, second OH, third OH. Just fill the hydrogen everywhere now. That's all is glycerol. This glycerol is also called as trihydroxyl propane. Trihydroxyl propane. See, trihydroxyl 1, 2, 3 and propane 1, 2, 3. Trihydroxyl propane. This right here is a glycerol. Right? Glycerol is colorless. It is viscous. Right? Colorless and viscous and everything. This is glycerol. That's all is going to be glycerol. That's all. Now, the question is very simple now. The question to all of you is very simple. Right? The question is, how are glycerol and fatty acids attached? Because in nature what happens is glycerol and fatty acids both are lipids. Both are simple lipids. These two simple lipids can actually join together. A glycerol and a fatty acid. Both of them can join together. Now how does it happen? How do they join together? Anyone knows the answer? Anyone? Students, smash the like button, all of you. First of all, freedom oil, nice freedom oil. How do they join? Anyone? How do they join? Two brothers. The magic is called as esterification. It's called as esterification. So glycerol and a fatty acids both of them can join by esterification. Okay, let me draw the diagram for you and show, because this usually they don't teach. It is given your NCERT, but many students and teachers also ignore this. Okay, because it requires chemistry, a little bit chemistry. All of you write down this one. Let's take a glycerol here. See, this right here is a glycerol. Now to this glycerol I will add a fatty acid. Okay. This is the R group. Okay. 14, 15, 16. 16 carbon, what do you call it? We'll call it palmitic acid. Okay. Now how do we join these two? Okay, understand. So when these two compounds join together, there is a release of water, right? There's a release of water. So H2O has been completely released and these two structures will join together. So this is what there is happening here is minus water is happening. Okay, and let's join these two now. So what do we get here? It's H2, right? H2COH. First one, okay. Hydrogen. Here we have oxygen is remaining. CH2OH. CH2OH. Okay, oxygen. And from here, this carboxylic acid will join. times CH3. Done. Now students, what did we do here? We took one of the hydroxyl group from here and we attach it to a fatty acid. So one out of the three hydroxyl groups here, one is replaced with a fatty acid. Now this structure right now becomes what? This structure right now becomes mono. Students, it becomes monoglyceride. It becomes monoglyceride. So when a glycerol plus fatty acid, when a glycerol and fatty acid, two different types of simple lipids join together, what do we get, students? Can anyone tell me in the chat? When a fatty acid and a glycerol, the two different types of simple lipids, when they join together, what do we get? Monoglyceride. This is given your NCERT, but they don't explain this entire thing, but my students should know. Okay, so formed by condensation of one fatty acid, monoglyceride. Now what if, students, if I add one more? What if I add one more fatty acid here? What do we get that time? Tell me. If I take one more fatty acid and I attach it here, then what do we get? Disaccharide, diglyceride. We get diglyceride. Okay, we get diglyceride. Now what if after the second one also, what if I take one more fatty acid and I attach it here? That time what do we get? Triglyceride. So monoglyceride, diglyceride and triglyceride. So what are we doing is we are adding more number of fatty acid group to a glycerol. Fatty acid to a glycerol with the help of esterification reaction is happening here. Okay. Now that was simple lipids. Compound lipids, only one example is given in NCERT. So write it down. This is the only example given here, fatty acid. Okay, only one example of compound lipid. Not fatty, compound lipid is given as lecithin. Okay, lecithin. So what exactly happens in lecithin? Esters of fatty acid and alcohol, that is basically glycerol plus fatty acid, along with additional group. So in the case of compound lipids, fatty acid, glycerol, additional compound is added. Okay, additional group is added. Okay, the additional group could be phosphorus, protein or sugar. Okay, the lecithin which we see here, right, lecithin. This lecithin has phosphorus attached to it. Don't believe me? I will show you. Let me show you. Can all of you focus here, students? Did we just now learn the triglyceride? Can you see here? Glycerol, one substitution, second substitution, third substitution. 1, 2, 3. Triglyceride. Okay. Now students, see, lecithin. Lecithin is a phospholipid. Now if you look at the structure of phospholipid, do you see here extra additional? This entire thing is the addition here. Can you see here addition, students, all of you? Phosphorus is added here. So that is a compound lipid. Apart from the glycerol, fatty acid, additional group is added. This is the additional group. Is added. That is a compound lipid. Okay. So there are different types. There is phospholipid, glycolipid, sphingolipid. These two are not given in your syllabus. This is the only thing given in your syllabus. Phospholipid. That is fatty acid plus glycerol plus phosphorus. Now students, do you want to know one small fact? Students, do you want to know a small fact? All of you. Do you want to know a small fact? No. See here, a fact is here. In your head, there is something called as white matter and grey matter. All of you are aware. White matter and grey matter. All of you know. Yes. But do you know what exactly is present in the white matter? The white matter of the brain is so called because it's whitish fatty appearance due to myelin. Right. So in your head, we have white matter. The reason it's called as white matter because it is white in color because of a fatty tissue, the fat lipid there. The lipid present in our brain, is a type of compound lipid, and it is called as galactocerebroside. It is mainly found in myelin. Okay, mainly found in myelin is the galactocerebroside. It is a type of glycolipid, compound lipid. Okay. So we learned about simple lipid, we learned about compound lipid. Under compound lipid, we learned about lecithin. Here. The last is called as derived lipid. Will not go detail of it. Example I'll tell you. The example of derived lipid in your NCERT is cholesterol. It is given in diagram. Cholesterol. Okay. From simple or conjugate lipids. Okay. This is cholesterol. Tetracyclic in nature. 1, 2, 3, 4. Tetracyclic in nature. Okay. Now students, with that we come to the end of lipids also. Now students, can we start something called as nucleic acids now? Nucleic acids. All of you are following now? All of you are understanding? Tell me, is this chapter easy or difficult? Difficult? We learned about amino acids. We learned about different types of lipids now. Very simple, very smooth. Okay. Now, let's start something called as nucleic acid. Very easy, very tiny concept also. Okay, very tiny concept. Now, students, the first question to all of you. Nucleic acids, are they part of retentate or filtrate? Go in the chat. Go quickly. Nucleic acids, are they part of retentate or filtrate? Okay. They're acid soluble or insoluble? Macromolecules or micromolecules? The weight is more than 1000 daltons or less than 1000 daltons? Weight is more than 1000 daltons. If they are macromolecules, they has to be a long polymer of something. What are the long polymer of? What are your nucleic acids a long polymer of? Nucleic acids are long polymer of, students. They are long. They are a polymer. They are a polymer of something called as nucleotides. When there are multiple nucleotides, join one after another, okay, phosphodiester bond, we end up getting the nucleic acids. Example of nucleic acid is DNA or RNA. Clear, students? Is this point clear to all of you? All of these points are clear to all of you? Yes or no? Yes. Now students, what is a nucleotide? This question arises, what is a nucleotide? Now I'll teach you the small basics of it. More you will learn in molecular basis. What is a nucleotide? A nucleotide is basically a monomer, I told you. Monomer of nucleic acid. Long chain of nucleotides will make up the nucleic acid. Okay. Gopika is also here, see. Gopika would have thought this in the molecular basis. Yes or no? Here are three main components. There's a phosphate, and then there is a pentose sugar, and there is a nitrogenous base. 3 different components make up the nucleotide. Now, what is a nucleoside? All of you, Gopika ma'am must thought you right. What is a nucleoside compared to nucleotide? Tell me in the chat. All of you. What is a nucleoside? What is a nucleoside? In this 3 components, pentose sugar, nitrogenous base, and phosphoric acid, one group is missing, right? What is missing? Yes, 5 marks to Gopika ma'am. 5 marks to Gopika ma'am. Ma'am, you take 5 marks. Enjoy. 5 marks. Additional marks. Additional marks. Right? Tell me. When nucleoside, right? The phosphate group is missing. Okay, nucleoside, the phosphate base is missing. So we only have nitrogenous base as well as pentose sugar in nucleoside. Now, students, we always learn, right? Okay. Again, one more question to all of you. What is the difference between DNA sugar and RNA sugar? Tell me how many of you answer this one. We always say, right, deoxyribonucleic acid and ribonucleic acid. What is the meaning of deoxy? What is the meaning of deoxy? Is there a difference in the sugar of DNA or RNA, or are they same to same? Let's see how many of you can tell. Let's see how many of you can tell before Gopika ma'am. Let's see. I'll give you 10 seconds. Quickly. What is the difference between DNA and RNA with respect to the pentose sugar? Let's see who can tell. Everyone is going blank now. I'll only tell off. Students, if you look at the sugar of DNA and the sugar of RNA, this is DNA sugar, this is RNA sugar. Here we call it as deoxyribonucleic acid, right? Deoxyribonucleic acid. What is the meaning of deoxyribose? The meaning of D means minus, something is missing. Oxy means oxygen. So oxygen is missing. Deoxy means D means missing, oxygen is missing here. Can you see? In the pentose sugar, in the second carbon, one oxygen is missing here. Right here. At the same time, if you look at the RNA sugar, okay, if you see in the RNA sugar, here we have in the second carbon, one extra oxygen is present here. Okay. Done? Yes. They answered. I didn't see, only they answered. Okay. Clear, all of you? So that is the basic difference between DNA sugar and RNA sugar. Students, there are so many lights sometimes, no? So many stars in the comment section. Because of all these stars, I become blind. What to do? Right? All these stars are making me blind. Right? So many stars in the chat. Now, next thing is called as nitrogenous bases. All of you know there is something called as ATGC. Right? ATGC, all the different types of nitrogenous base, and also uracil as a nitrogenous base. Okay. Now here, there is something called as purines and something called as pyrimidines. Now, how do you remember purines and pyrimidines? Right? One small, this thing, the trick is there. Okay, a small trick. For purines, it's, which I learned long back, it is gold is pure. Okay, gold is pure, that is guanosine, gold as pure. A is going to be adenine and P is going to be purine. Gold as pure. And for pyrimidines, that is cut pyramid. What is cut? Cytosine, uracil, thymine, P for pyrimidines. Cut the pyramid. That's all. Okay. That's all students. Okay. So pure as gold as well as cut the pyramid. Very simple. I don't think you can go wrong here. If you remember the small trick, you can remember the what is different types of pyrimidines and purines. Now one question arises here, right? One question arises. which of the following are double ring structure? One question is there. Which of the following of nitrogenous bases are double ring structure? The answer is very simple, students. If you look at the purines, they are double ring structure. Can all of you notice? 1, 2, 1, 2. They are double ring, the purines. But if you look at the pyrimidines, they are single ring. They are always single ring structures. Pyrimidines. Small difference, remember that. Okay. Now, purines, these two, pyrimidines, all the three. Cut, C-U-T, gold as pure. Okay. Done. Nucleotide will have all three. Nucleoside will only have sugar and nitrogenous base. Phosphodiester bond, students, I will not teach you the detail of it right now because all of you would have learned it in the molecular basis, because I think ma'am has drawn the entire structure there and taught you there. But remember, phosphodiester bond is a bond present between the two different nucleotides. One nucleotide, second nucleotide. They are joined together with the help of phosphodiester bond. 3' to 5'. Can you see here? 3' to, this is a 3' here, to 5'. 3' to 5', phosphodiester bond can be present here. Phosphodiester bond. DNA is double stranded, RNA is single stranded. Okay. Nitrogen bases we have here ATGC, here we have uracil instead of thymine, AUGC. Okay. Now students, if you see here, we learned about fatty acids, palmitic acid, students, tell me, 16 carbon. We learned about glycerol, we learned about triglycerides, we learned about glycine, alanine, serine today, all different structures. We learned about glucose and ribose sugar, yes or no? Lecithin is a phospholipid. Cholesterol is a derived lipid right here. Nucleotide, students, always remember, nucleotide will have ic acid, IC acid, nucleotide. Nucleoside will be uridine, adenosine, thymidine, INE is going to be your nucleoside. While nucleotide is going to be ic acid. Acid is going to be for nucleotide. Remember that. Purines, pyrimidines. It's given here. Remember always, purines, adenine is a double ring structure. 1, 2 ring structure. Pyrimidines are single ring structure. Okay. Now students, that was the end of the nucleic acids. Very simple, very basic it is. Next thing is called as metabolites. Okay. Now students, understand a plant, right? Do you know there are certain plants which can produce toxin, and once they produce the toxin, no other herbivore will come and eat it. Alright. Can anyone give me a certain example? Can anyone give me a certain example of a plant which can produce a toxin which if a cow or a goat come and eats it, the cow and goat will never come and eat again? Anyone? Cardiac glycoside. Any plant which can produce cardiac glycoside? It is given in your NCERT in the ecology. In the ecology we learned, remember, Calotropis. Absolutely correct. All of you smart now suddenly. All of you remember Calotropis. Absolutely correct. Calotropis is such a plant which can produce certain type of toxins. Now remember students, apart from Calotropis, there are other plants which can also produce different types of substances which are not directly involved in plant life cycle. They are called as secondary metabolite. Okay. See here. There is something called as primary metabolites involved directly in growth. Carbohydrates, lipids, proteins, nucleic acids, direct involvement. But there are certain products produced by the plants which are not directly involved for defense purposes, stress purposes, and these are different examples given here. Now here is a table given to you in NCERT. Let me tell you, you have to by heart this table at any cost. At any cost, you have to remember this table at any cost. Okay. They are the secondary metabolites. Pigments, carotenoids, anthocyanin, alkaloids, morphine, codeine. Morphine is given in that term. You might have seen drugs. Morphine. Then there is codeine. Codeine is in the cough syrup. terpenoids, monoterpene, diterpenes, essential oils. Nice smell, you get a nice smell. Essential oils. Toxins, arbutin and ricin. Lectins is concatenatin A, which is also a growth promoter. It's a type of cell division promoter actually. Then there is vinblastine and curcumin. Vinblastine is a cancer curing agent. Curcumin from curcumin longa, it is turmeric. Rubber, gum, cellulose. All of these are different types of secondary metabolites. You have to remember it at any cost. You have to remember. Okay? Because you will get a lot of questions. You will get a lot of questions on that. Okay? Students, with that, let me tell you, half of the chapter is over. Half of the chapter is over right now. Right? Now you might be wondering, sir, it took us almost 2 hours to reach here. All of you agree? Right? It took us 1 hour 15 minutes to reach here. Right? 1 hour 15 minutes to reach till here. Right? Now let's take a 15 minutes break. And I want you to refresh your mind and come back. I don't want you to burden yourself with so much information. I know you have learned a lot right now. We learned about secondary metabolites, amino acids. We learned about nucleic acids. We learned about lipids. We learned about amino acids. Right? So we have learned a lot right now. Half of the chapter is over. Now I will give you 15 minutes break. 15 minutes break. That's all. Not more than that. Because I want you to go freshen up your face, wash your face nicely, drink some water, eat something and come back. Okay. After you come back, we'll go to the second part of the chapter, which is actually more important. Let me tell you now only. Second part is actually more important than the first part. That is my guarantee. Because in the second part, I will be teaching you carbohydrates and most importantly, the enzymes. The enzymatic reaction. What is Vmax? What is half Vmax? What is a cofactor? What is the apoenzyme? What is a holoenzyme? What is the inhibitor? Competitive, uncompetitive, non-competitive. So all of these things in the second half. Okay. So what is the time now, students? Let's take it as 1:12, 1:25. Only will take it off. Okay. So until 1:30, all of you can eat something for 15 minutes, aram se chill, and then come back. Okay. Once you come back, we'll finish the rest of the chapter for 1.5 hours more, 1.5 or 2 hours more. Okay. So with that being said, thank you so much. I'll see you in exactly 15 minutes. Okay. Sound check, sound check. Now students, let's start with something called as carbohydrates now students. Quickly, let me know the start students. Let me in the chat know right now if I'm audible and visible to all of you, because after break sometimes what happens is the mic stops working. So quickly let me know in the chat right now if I'm audible and visible to all of you. Okay. Now, let's start with something called as carbohydrates. Okay, carbohydrates. First of all, students, how many of you love eating rice? Tell me in the chat. Like you love eating curd rice, biryani, or anything related to rice item, because this is the carbohydrate which you take in, right? So what are carbohydrates? Carbohydrates are nothing but hydrates of carbon. Hydrates of carbon. Here we have different types of structures which has multiple OH groups, and they are basically hydrocarbons. They have hydrogen and carbon in them. Now the famous example here is the glucose, which all of you are aware, C6H12O6. Apart from glucose, there is also fructose. So glucose and fructose are the two different examples of carbohydrates given your NCERT. Okay, carbohydrates. Now apart from that students, every single carbohydrate will have something called as aldehyde group or a ketone group. Okay. Now based on the number of monomeric units, carbohydrates can be divided into multiple different categories. For example, there is something called as monosaccharides, disaccharides, oligosaccharides and polysaccharide. Now what is a monosaccharide? Understand. Monosaccharides are the most simplest saccharides. They cannot be divided any further, and they are made up of a single monomeric unit. Okay, single monomeric unit, simplest carbohydrate. Now what is a disaccharide? Disaccharides are different types of carbohydrates where we have two different monomeric units which are joined together with the help of glycosidic bond. For example, sucrose is made up of glucose plus fructose. Two different monosaccharides are joined together to form a disaccharide. Lactose, we have glucose plus galactose. Two different monosaccharides are joined together to make up the disaccharide. So disaccharides basically have two monomeric units joined together. The link is called as glycosidic bond here. Okay, link is called as glycosidic bond. Done? Done. Now, what is oligosaccharide? Oligosaccharide basically means 3 to 9 monomeric units. But what are we interested in? We are mainly interested in the polysaccharides, which are mainly the macromolecules which are present in acid insoluble fraction, retentate, whose molecular weight is more than 1000 Daltons. So all of these are polysaccharides. Okay, they are polymers of monosaccharides with 10 or more monomeric units. So there are basically 1, 2, 3, 4 long chain of monomers, and all of them are joined together with the help of glycosidic bond. Now here students, how will the question come? Here the question will come based on the classification. Okay, there is something called as homopolysaccharide, homopolysaccharides, as well as heteropolysaccharides. Heteropolysaccharides. Now what is the difference? Understand. In the case of homopolysaccharides, they are made up of same type of monomeric unit. For example, long chain of glucose, long chain of fructose. So the monomeric unit is going to be same to same throughout. Okay. Now here, this can be further classified into two main categories. Okay. The first category is based on the structure. Certain polysaccharides used in structure, certain polysaccharides which are used for storage. Okay. For structure, we have, in the case of plant, we have cellulose. In the case of animal, in the case of basically in the insect and all, we have the chitin. Okay, insect chitin. Now, under storage, again we have three types. One is present in the case of plants, that is starch. Second type is also found in the case of plants, that is called as inulin. And the third type, which is present in the case of humans or animals, that is called as glycogen. So what did we learn right now? We learned that under homopolysaccharides there is for structure as well as storage. The example for structure is cellulose and chitin. For storage, it's going to be starch, inulin as well as glycogen. Okay. Now under heteropolysaccharide, there are two different examples here. Understand. First one is called as peptidoglycan. Second one is called as agar-agar. Okay. Peptidoglycan is made up of NAG and NAM. NAG and NAM are two different types of repeating units here. NAG and NAM. NAM and NAG. Okay. N-acetyl muramic acid, N-acetyl, right, galactose. So two different types of monomeric units which are repeating in the case of peptidoglycan. In the case of agar, there are again two types are there. So what are the functions here? All of us are aware. See, this is the storage. This is the structural. In structural, we learned about cellulose. It is a polymer of glucose, long chain of glucose, unbranched, used for paper industries, right? Chitin, polymer of N-acetyl glucosamine. It is present in the structures of the insects, the exoskeleton of insects. Then under storage, we learned about starch, polymer of glucose, glycogen, plain polymer of glucose, but inulin is a polymer of fructose. So that is very important. So what is the polymer made up of? Right. So here it is made up of fructose, inulin, glucose and glucose. All three are storage polysaccharides present in animals and plants. Okay. N-acetyl glucosamine, N-acetyl muramic acid, NAG and NAM, that is peptidoglycan, two different. Agar-agar, again two different. They'll not ask you this, they might ask you what is a heteropolysaccharide. Examples they might ask you. Okay. Now this next concept called as a reducing agent. Right. We might have heard about reducing agent in your chemistry. Right. We usually learn as your polysaccharides as well as your disaccharides and as well as monosaccharides. We tell that some of them are reducing agents, some of them are non-reducing agent. Now, what is the basic difference and on what criteria are we deciding it? The answer is very simple. The availability of free aldehyde or a ketone group. Okay. See here. If you look at the monosaccharides, they have a free aldehyde here and a free ketone group in the case of monosaccharides. But in the case of disaccharides, they do not have free aldehyde or ketone group, not present at all. So, most of the aldehyde and sucrose are non-reducing, while your reducing sugars are mainly monosaccharides. Monosaccharides are reducing sugar. Now, how do we find if it's reducing sugar? There is a small test. Okay, what is small test? The small test is called as Benedict solution. Benedict test. What you do is, in this test tube, take 2 ml of glucose solution. Equal amount of Benedict solution is added. This is the test tube right now. Now, once you heat it, there is a color change. Now, what exactly happened here? Right. Basically, all the cupric become cuprous ions, so they became, and there is a change in color. So, if there is change in color, it means it is reducing sugar. it is reducing sugar. If there is no color change, non-reducing sugar. Okay, very simple it is. See, reducing sugar reduce the cupric ions to cuprous. Cupric become cuprous. Change in the plus. These are non-reducing sugar. Can you see? Non-reducing sugar. They do not have any free aldehyde, do not have any free ketone group, not present at all. Non-reducing sugars. Okay. Now there is one more experiment called as iodine test. Now what is the significance of this test? Very simple. Okay. See here. For example, in one test tube you are taking starch. In one more test tube you are taking cellulose. Starch here, cellulose here. Both are polysaccharides. Both are polysaccharides. But in the case of starch, it is showing blue color when you add iodine. When you add iodine to the starch, it is showing blue color. But when you add cellulose iodine, it is showing brown color. Now, what is the difference here? What is the difference? The answer is very simple. The answer lies in the structure of it. Now, let me ask you a simple thing, okay? You are standing somewhere there, okay? Now, if I take this pen and if I throw this, and if I ask you to catch, will you catch? Tell me yes or no in the chat, tell me. If I take this pen and I throw this pen at you and I tell you catch the pen, will you catch? You will catch. Okay. Now what if I am tying your hands, right? What if I tie your hands and I take the pen and throw the pen at you? What will you do? Can you catch right now? Will you try to catch with your mouth? No. The pen will fall on your face. It will fall on your face. In the scenario 1, your hands were free. You were able to catch the pen. In scenario 2, I tied your hands. You are not able to catch the pen. Now apply the same concept here. In the case 1, what is happening? The starch has something called as branches. Can you see? The starch has branches. Just like your hand is free, it has branches. These branches can hold the iodine solution. They can hold the iodine solution. But look at the structure of cellulose. If you look at the structure of cellulose, there are no free hands here. No free hands. Tell me, if there are no free branches here or no free vesicles here, can it trap the iodine? Not possible. See here. Cellulose does not contain complex helicals, hence cannot hold I2 molecule. Starch can hold I2 molecules in its helical portion. So it has helical portion here. It can hold, so positive. It cannot hold negative. That's all. Very simple concept. Like if I tie your hands, you cannot hold the pen. You cannot catch the pen. Okay. Student, the answer is given already here. The question says like this: cellulose does not form blue color with iodine because it does not contain helical complexes, hence cannot hold I2. This is all right here as a NEET PYQ. Students, answer is given. What is this? Mistakenly answer is given, students. I didn't check it. Okay, see here. All of you know inulin, all of you know. All of you know this also. Exoskeleton of arthropod is composed of chitin. But mistake answer is given, students. But mistake answer is given. So any doubt now, students? Any doubt in carbohydrates? The only thing which you need to learn in carbohydrates is the structure. What is homopolysaccharide? What is heteropolysaccharide? These two things are the main thing here. Okay. And examples which you need to remember for each one of them. And with that, it is over. Now I'll teach you something for proteins. We have learned before, right? All of you remember this? 20 different types of amino acids are there. We learned that. We learned about essential and non-essential. We learned that also, right? Now here is a different functions of protein. For example, there are multiple functions of a protein which you need to know. Because enzymes are also protein. Yes or no? See, example is given here. Trypsin is an enzyme, is a protein. Then we have collagen. It forms intracellular ground substance in the body. Then there is insulin, which is a hormone, is also a protein. Yes. Then there is antibiotics, are also proteins. See, fight infectious agents. The protein. Some proteins can act as receptors, help in recepting or help in picking up signals. Some of them can be transporting agent. Example is GLUT4. Enables glucose transport into the cell. For example, if glucose cannot enter inside, with the help of GLUT4, it can enter inside. So these are different types of proteins and their functions. This table you have to remember at any cost. You have to remember. Okay, all of you will remember, right? You have to remember. Now there are two statements here. Remember that also. See, first is collagen is the most abundant protein in the animal kingdom. Collagen. If they ask you which is the most abundant protein in animal kingdom, it is collagen. If they ask you which is the most abundant protein in the entire biosphere, that time answer is rubisco. That time answer is rubisco. Right? Rubisco is the most abundant protein in the whole biosphere. All of you remember? You have to remember. Okay. Now students, we finished polysaccharides also. We finished today. Yes, we finished. Okay. We learned about starch. We learned about inulin is a polymer of fructose. We learned that. Cellulose is a homopolymer. We learned that. And heteropolymer. We learned this also. Why you cannot hold I2. We learned that also. We learned about this one line is here. See, understand. In the polysaccharide chain, say glycogen, the right end is called as the reducing end. So remember students, RR. In a long polysaccharide chain, right end is called as reducing end and left end is called as non-reducing end. Reducing end. Okay. We learned this also. Paper we learned about. We learned about the complex polysaccharides, right? Example is the chitin, N-acetylglucosamine. Chitin is a complex polysaccharide. Yes. We finished nucleic acid also. We learned about nucleotide. We learned about nucleoside, macromolecules. We learned about the nitrogenous bases, pyrimidines and purines. Yes, we did that also. We learned about DNA and RNA. Done. Now, one small question. Just solve this question. Tell me the answer in the chat. How many of you know the answer? Tell me answer in the chat. Which of the following is the most abundant protein in the animal kingdom? Simple question, simple answer. Quickly. Quickly answer in the chat. Done. Animal kingdom, it is collagen. Now till here was the basic things which you learned till now. We learned about carbohydrates, we learned about nucleic acids, we learned a little bit of proteins and their functions, we learned about carbohydrates, we learned about amino acids, and we also learned about lipids. Okay, that was the main 70% of the chapter is over, and I believe till now there should be no doubt we have tackled every single thing till now. Now we go to the next part of the chapter where many questions usually arise. For example, we're going to start right now the enzymes part. Okay, before we go to the enzymes part, there's a small concept called as structure of protein. So let's identify that and understand that, and then we go to the enzyme part. Okay, can we start? Yes. Now this is the structure of protein. Now a protein has multiple levels of structure. There's primary, secondary, tertiary and quaternary structure of protein. Now for example, if you look at this diagram given your NCERT, here it says primary structure of protein. Now all of you should remember, a primary structure of protein always tells you what? What is the significance of primary structure of protein? That you need to remember. First of all, remember primary structure of protein is always linear. Can you see? It is always linear. It is always linear in shape. Now when it is linear, now what is the significance? The significance of primary structure is that the primary structure of protein tells you which amino acid is present at which location. I'll repeat again. Primary structure of protein tells you which amino acid is present at which location. For example, if you notice here, first amino acid, second one, third one, fourth one, fifth one. Okay, let's say here I have leucine, here we have lysine, here we have arginine, where we have methionine. So which amino acid is present at which location is told by primary structure. And in primary structure, we mainly have the peptide bond. We have the peptide bond. Peptide bond is a bond between two different amino acids. Peptide bond. Okay. Then we have secondary structure of protein. Secondary. In secondary, we have two types. One is alpha, other one is beta. Alpha helix, beta pleated. In these two structures, this one is like a staircase. You might have seen, right? The round staircase, solenoid model in physics you might have seen. That is how protein looks like. Or a protein can look like this. This is called as beta plated. Let me tell you what is beta plated. All of you see this. For example, this is the paper. This is the paper. Okay. Now, what if I fold it? Can all of you see this? Can all of you see this zigzag pattern? This is called as beta plated. This is called as beta plated sheets, and that is how protein is arranged. Can you see beta plated sheet? Now here in the case of second structure protein, we also have something called as hydrogen bonding can be observed here. Hydrogen bonding. Okay. Now next is tertiary structure. Now in tertiary structure, alpha and beta both are fused here. Both are fused here, and the entire structure looks like a round ball. Can you see? The entire structure right now looks like a round woolen ball. This is called as a tertiary structure protein. Now this is 3D in shape. It is 3D and it is also biologically active. It is 3D and biologically active. Okay, 3D and biologically active. Here we also find disulfide bonds can be present here. Last is called as quaternary. In quaternary, what happens is, for example, this one is present here. Here, this is present here. Similarly, there is one more tertiary which will fit here. Which will fit here. Okay. So in quaternary, what do we have here is multiple tertiary will make up the quaternary. Here we have subunits. Subunit 1, subunit 2. Two subunits of tertiary is coming together and making the quaternary. Let me give a small recap. In primary, what we learned? Linear chain of amino acid. Linear chain. Here we have right side is called as C terminal, left is called as N terminal. N. Why is it called as N terminal? Because here we have free amino group. Why is it called as C terminal? Because we have free carboxylic acid here. C terminal. Okay. Represents position of amino acid. Which amino acid is present where? That is told by the primary structure. Primary structure. Okay. Now secondary, alpha and beta. This is alpha. Alpha helix model. Alpha helix. This right here is beta plated sheets. This is how second structure. Tertiary structure. Three-dimensional. Protein is folded upon itself. Biologically active. It is going to be biologically active here. Tertiary structure protein. Okay. Quaternary. See here. In quaternary, what happens? Two or more polypeptide chains. So we have multiple polypeptide chains joining together here. One from here, they are joining together. Okay. Each chain is called as subunit, I told you. Subunit 1, subunit 2. Okay. Arrangement of each folded polypeptide chain with respect to each other are present here. The example is hemoglobin. Read RBC hemoglobin. The protein hemoglobin has 4 chains. 2 alpha chains and 2 beta chains. See, this is a example of your hemoglobin. Hemoglobin has 1, 2 beta chains, 2 alpha chains. So this is a quaternary structure. In this quaternary structure, there are 4 different subunits. 2 beta, 2 alpha subunit. Okay, they are coming together, they are joining. Okay. Primary, secondary, tertiary. You can read it, same thing is given here. 3-dimensional view, structure is absolutely necessary for the primary biologically active proteins? Quaternary structure. Okay. So that was the protein structure. I hope all of you are able to follow the protein structure. Very easy to understand. Primary, secondary, tertiary, quaternary. Tertiary onwards, it is 3D in shape. Tertiary is biologically active in nature and it is like a woolen ball. Quaternary, multiple peptides joined together. Okay. Now, there is different type of bonds in this chapter. For example, there is something called as peptide bond. What is peptide bond, students? Peptide bond is a bond present between two different amino acids. Then there is glycosidic bond. Glycosidic bond is present between two different carbohydrates and monosaccharides. Okay. Now, one small question here. Let's see who can solve this question. Let's see who can solve this question. Small question here. We just now learned protein has which type of bond? Protein has, tell me students, protein has peptide bond. Unsaturated fatty acids will have what? Double bond, I told you already. Okay. Nucleic acids will have phosphodiester bond. Polysaccharides will have glycosidic bond. D is 3, A is 4, B is 1. C is going to be 2. So option number 3. Option number 3, yes or no? Okay. Now, sir, there are different types of protein. Structural, contractile, nutrient. We learned, right? Regulatory, defense mechanism, antibiotics, transport, GLUT4, catalytic. So there are different types of protein, right? Now, out of all of these different types of proteins, our main focus has to be on the catalyst part, because that is what is the given in NCERT, and that is also is the most important property of a protein. That is, certain proteins can become catalytic in nature, right? They can provide enzymatic reactions. Okay. So what is a catalyst? From here onwards, we are starting with the catalyst. Okay. Can anyone tell me the definition of a catalyst, students? With respect to chemistry or general science, what is a catalyst? Anyone? What exactly is a catalyst? Anyone, students? All of you quickly smash that like button. See, catalyst and enzymes, yes, they are same. But what exactly is a catalyst? Catalyst is a chemical substance. It is a substance which can alter the rate of reaction. It can alter the rate of reaction. It means that it can increase the rate of reaction and also decrease the rate of reaction. Both can be done. So it can increase or decrease the rate of reaction. Both can be done by catalyst. There are two types. Mainly there are two types of catalyst: inorganic and organic. Inorganic catalyst as well as organic catalyst. Now, what is inorganic? Inorganic are used in your chemistry. For example, platinum. Yes or no? Platinum is an example here. Now, what about organic catalyst? Example is the enzymes. It is the enzymes. So, enzymes are basically organic catalyst. Like inorganic catalyst, platinum. Okay, remember the simple difference. Okay. Now we come to enzymes. Now, what are enzymes? Enzymes are basically biocatalyst. They're basically biocatalyst. They are organic in nature. Yes, and all enzymes are proteins. All enzymes are proteins except the ribozyme. Except the ribozymes. Ribozymes are RNA enzymes. They are the RNA enzyme. Clear, students? Clear. Now, students, remember, all of these enzymes are highly specific in nature. They're highly specific in nature. They play lock and key. You cannot use any random key to open any lock. Every lock has a specific key made for it, or every key has specific lock for it. Only with the help of that lock, the key will, only with the help of that key, a lock will open. They are very highly specific, just like that, lock and key. So here also they are very highly specific in action. Highly specific in action. Okay, highly specific in action. They need aqueous medium to always react and everything. Okay. Now, students. Now, what is an active site? Understand what is an active site. Now, students, do you remember the tertiary structure? Students, do you remember the tertiary structure of protein? All of you remember? Like a woolen ball like structure? Same thing. Now, students, in this tertiary structure of protein, there are certain gaps. Can you see here? There are certain gaps or crevices, or we can call it as pockets. Okay, I can call it as pocket or crevices. Now these crevices or pockets, in these crevices and pockets, there are mainly certain functional groups present here. And onto this functional group, our substrate will come and bind. What will come and bind? Substrate will come and bind. Now I'll give you a simple example. Okay. For example, all of you have seen a lock. Okay. Now can you take a key and can you open the lock at any random place? Is it possible to open the lock at any random place just because you have the key? Can you keep it anywhere, like in abracadabra somewhere? If you keep and tell a magic spell, will the lock open? Not possible. We have to take the key, put it inside the hole in the lock. Now, this right here is the hole like that. On the lock, just like how in the lock we have a small hole, on this enzyme, there is a small hole there. And that small hole where the lock key will come and attach, the small hole is called as active site. It is called as active site. All of you understood? What is an active site? Active site is the point on the enzyme, not on the substrate. It is the point on the enzyme where your substrate will come and bind. Okay, like how on the lock we have a small hole. Lock is the enzyme. Okay, on this enzyme we have this small point here where the key will come and attach, or a substrate will come and attach. Okay, substrate is the key. Imagine substrate is the key and this enzyme is the lock. Enzyme is the lock. Now students, you might be wondering how efficient the enzymes are. Like, are they really that great? Are they really that amazing enzymes? Or can we survive without enzymes? Let me tell you how important enzymes are. Like, what is the actual magical property of an enzyme and how good enzymes are. For example, this is a simple reaction here. Carbon dioxide plus water, carbonic acid. Simple reaction. Now this simple reaction without catalyst is happening here. Okay, without catalyst it is happening here. Without catalyst, what is happening? 200 molecules of H2CO3 is produced. It is produced in 1 hour. In 1 hour, we are getting 200 molecules of H2CO3. But if you add a catalyst, like carbonic anhydrase, now is there any change in the rate of reaction? Will the reaction happen very fast or slow? You can't even imagine how fast it happens now. I will give you a perspective now which will blow your mind away. Amazing perspective. Listen to me. If you add the catalyst, we are approximately getting 6 lakh molecule of H2CO3 in. Here 200 molecules in 1 hour. Do you think we are getting now 6 lakh molecules in 1 hour? Okay, not 1 hour. In half an hour? No, less than that. 10 minutes, 6 lakh molecule? Less than that. 5 minutes? Less than that. 1 minute, 6 lakh molecules? Less than that. In 1 second. In 1 second, right? Do you realize that 200 molecules in 1 hour without catalyst, but as soon as you add catalyst, 6 lakh molecules of H2CO3 in 1 second, per second, 6 lakh molecules you're getting. That is the power of a catalyst. Okay, it's given here also. Now here they have given a formula for the rate of reaction, right? Rate of a physical or chemical reaction is the amount of product formed per unit time, that is delta P by delta T. Okay, if you give a direction, you can find the velocity, it can be called as velocity. Okay, now there is a general rule here. Every single time there is a reaction happening, if you increase the temperature, reaction will happen faster. Decrease the temperature, reaction will happen slower. Simple. Okay. Now students, let's understand what is enzymes now. No, enzymes will come later. No. How does it actually happen? How does it actually happen? I told you catalyst has amazing superpower, but how does it actually happening? Like how is the catalyst even making this entire amazing thing even happen? How is it even possible? That we will learn now. Okay. Now first understand a simple chemical reaction. What happens? We have a reactant 1, reactant 2, and we get the product. But this entire reaction is not very simple. Entire reaction is not so simple. Like 2 reactants are joining, getting product. No, it is a little more complex than that. Okay. Now what is happening is when one reactant is reacting with other reactant, we end up getting a middle phase. The middle phase is called as intermediate product. It can also be called as transition state. So before we get the products, in the middle there is an intermediate product that is called as transition state. Okay. So when 2 reactants, before we get a product, there is transition state. Transition state, right? Transition state or intermediate product before the product is formed. Now generally what happens is, listen to me very carefully. This intermediate product, right? It has very high energy. This one transition state has a very high energy. Okay, very high energy. Now understand. Now what is the meaning of activation energy now? This you generally learn. If you learn this, the entire concept is going to be very easy to you. If you understand what is activation energy. Now just now I told you there is reactants, product, in the middle there is a transition state who has very high energy. Some energy here, some energy here. Okay. Now understand the definition. Of activation energy. It is the average energy difference. Between substrate and transition state. So there is something called as activation energy. It is the energy difference between the substrate and the transition state. Obviously, the energies are not same. Transition state has very high energy and substrate has lower energy, right? So there is a difference in energy. The difference in energy, or the average difference in energy, is called as the activation energy. Let me show you. See this, all of you. Here is the substrate. Substrate is here. Here we have transition state. Here we have transition state. Listen to me very carefully. Can you see the potential energy here? Potential energy here. Some energy is present here. Some energy is present here. Now there is a difference in energy. Can all of you see? There is a difference in energy. Difference in energy. The difference in energy between substrate and transition state. The middle part is called as what? It is called as activation energy. Now, what is enzyme doing actually? What is enzyme doing? Let me tell you a small scenario. Okay. Let's take one character called as Raju here. Okay. Here we have Raju. Now, this Raju has to jump over this wall. This Raju is coming, running and coming. Okay. Let's make the wall even bigger. Okay. If the wall is this big. Okay. Now, students, this Raju has to climb the wall and he has to come this side. Raju has to come this side. Now here I have given him a, let's say there is a ladder also here. There is a ladder to climb this. There is a ladder to climb this. Okay, there is a ladder to climb this. Raju is going. He is climbing a ladder, going this side, coming down and coming here. Now it is taking him, let's say how long will it take? It is taking him 30 minutes. 30 minutes to do this exercise. 30 minutes. Okay. Now this, can you see? This large thing, can you see this energy gap? This is called as the activation energy. It is also called as your activation energy. Now what if I bring one bulldozer? What if I bring one bulldozer and I break this wall? I break this wall. Now here we have Raju. This is case 2. This is case 1. In case 2, Raju is going. But right now, the wall is this much. Here we have wall this much big. Here we have wall this much. Now can the Raju easily go this side? He can go this side. How much do you think it will take him? It will take him hardly 30 seconds. Maximum 30 seconds. Maximum 30 seconds. It will take him 30 seconds. Now do you realize? Activation energy was there. Entire activation energy is decreased. Entire activation energy is decreased here. Entire activation energy is decreased completely compared to here. The same thing is happening in the case of your catalyst also. Now can all of you notice here? There is something called as activation energy. Once you, without catalyst, can you see? Activation energy without catalyst. Large hump is there, like a large wall. Now this substrate has to become product. It has to spend a lot of time reaching that energy. A lot of time is thing. But as soon as we add the enzyme, what is it doing? Enzyme is decreasing the activation energy. Enzyme is decreasing the activation. Like here, how this entire activation energy was decreased, like this decreased, right? Same way, here also, enzyme is decreasing the barrier. So this is be able to convert to product very easily. That is all is the concept. How many of you understood the concept? Red hearts in the chat. Red hearts in the chat. How many of you understood the concept of activation energy? Activation, the energy difference. Decrease the energy difference is done by the, it is done by the enzyme. It is done by the enzyme. Understood all of you? Understood? All of you? Yes or no? So enzyme is decreasing or reducing the activation energy. So what exactly is happening? Reactant 1, reactant 2, enzyme is added, we get the product. If the activation energy was this much, now this will be reduced to somewhat like this. Somewhat like this. Because of enzyme. This is with enzyme. With enzyme. With enzyme. There's a small thing here which we have to learn, the entire steps. First we have the substrate plus enzyme. Then we get enzyme substrate complex. This enzyme substrate complex will become enzyme product complex. Then it will get converted. Enzyme will be separate, product will be separate. This is the entire reaction which happens. Entire reaction will happen. Okay. Don't worry, I will do cell cycle again. Don't worry. It is happening this week only. Cell cycle is happening this week only. Don't worry. Okay. Done. All of you understood? Enzyme substrate complex, enzyme product complex. Product and enzyme are separate later on. Now there are two types of reaction. All of you are aware. There's something called as endothermic, exothermic. But with respect to the energetics, how do we understand endothermic, exothermic? Depending upon the energy, listen to me. In the case of endothermic reaction, can you see? Reactant energy is very less. So what do? In the case of endothermic reaction, do we absorb energy or do we release energy? Students, tell me. In the case of endothermic reactions, the reactants will absorb energy or release energy? They'll absorb the energy. So, reactants are less energy. They will absorb more energy and they get converted. Endothermic. Now, what about exothermic? In the case of exothermic reaction, reactants have higher energy. So, what will they do? They will release energy. And then they'll become products easily. So endothermic is absorption because the reactant have less energy. Exothermic reactants have more energy coming to products. So they release some energy and they become products. Okay. Very simple. Catalyst and enzyme. I told you, right? Catalyst is a inorganic. Catalyst is a general term for an enzyme. Enzyme, when we always say enzyme is always related to biology. Catalyst can survive at high temperatures. Catalyst works at high temperatures. But enzymes generally cannot work at high temperatures. Except a few. Thermus aquaticus one. Right? Taq polymerase. Except Taq polymerase, enzymes cannot work at high temperature. They will denature. Okay. Enzymes are organic in nature. Right? Like trypsin, pepsin. Catalysts are inorganic in nature, zinc and everything. Okay. Now here is a simple thing which we learned right now. The catalytic cycle of enzyme action can be described as follows. First, what happens? The substrate binds to the active site on the enzyme. I told you, it's always substrate will come and bind. The substrate will come and bind here. That is a lock. That is the key. Sorry, key. That is the key. Right. Bind to the active site on the enzyme, fitting into the active site. Okay. Active site is the hole. Enzyme is the lock. So what is happening? Substrate is coming to the hole on the enzyme. Okay, lock. Fitting. The binding of the substrate induces the enzyme to alter the shape, fitting more tightly around the enzyme. So as soon as the substrate is inserted, the entire enzyme will go under some changes. It will nicely hold the substrate. The active side of the enzyme now is close proximity of the substrate breakdown and chemical bonds of the substrate, and the new enzyme product complex is formed. Okay. One small question here. In a reaction catalyzed by the enzyme, which of the following statement is correct? Students, first answer is only correct, no? Enzyme decrease the activation energy for formation of transition state. First one is correct. First itself is correct. They will decrease the activation energy. Okay. There are multiple factors affecting the enzyme reaction. There's temperature, pH, enzyme concentration, substrate concentration, inhibitors. All of these are different types of factors which influence the entire rate of reaction. Okay. Now one by one we'll tackle every one of them. One by one we'll tackle now. Okay. Understand. First one is temperature. Now, if an enzyme has to work, it needs a proper temperature. If you increase the temperature, proteins will completely become denatured. If you decrease the temperature, proteins will freeze up, enzymes will not work at all. So there should be an optimum temperature at which the enzyme will work. Optimum temperature. Okay. Same goes for pH also. Same goes for pH. pH there is an optimum pH at which an enzyme will work. And every single enzyme has a different optimum pH. Like a trypsin will have different, pepsin will have different. Every single enzyme has a specific pH and temperature at which they'll work. Okay. Now what is substrate concentration? Understand, very simple concept. Very simple concept. Okay. Now tell me students, what is your favorite food? What is your favorite food? I'll teach you this one with the help of food only example. Tell me, what is your favorite food item? Simple example, tell me. What is your favorite food item? I will take one from the chat only today. Because this one is very difficult to understand, but I'll make it easy for you. Tell me, what is your favorite food item? Last time also I gave biryani example. Tell me something else apart from biryani. Apart from biryani, anyone? Any other example? Biryani only, everyone. Okay, fine. We'll take biryani example only. Okay, we'll take biryani example only. Listen to me very carefully. Okay. Now here, let's take you are the enzyme right now. Let's take you are the enzyme right now. Okay. So enzyme is here, enzyme is here, enzyme is here. Okay. Now initially I am making you all sit in front of me and I'm making you starve. Okay. All of you are very, very hungry right now. All of you are very hungry. You're sitting in front of me and I'm giving you food. Let's say I'm giving you biryani today. It's called biryani theory. Okay. I'm giving you biryani. Okay. Now initially what happens? All of you are very hungry. You're hungry. What are you? You are enzymes. All of you are enzymes. Now what do? I will give you substrate. I'll give you substrate. Okay. Will your stomach become full? Obviously. Well, if I keep giving you biryani, let's say I gave you one full plate biryani. You're like, I'm full now. Now, what if you're still hungry? Some of you will be still hungry. One plate is not enough. I'll give you a half plate more. Can you see? Some of you will be full. Some of you will still be empty. Now, what if I give you 10 kg biryani now? Can you keep eating more? Or will you become saturated? Tell me. If I give you 10 kg biryani more, will you keep on eating or will you become like, you'll start like, I'm done, you'll start crying. I'm done, you'll say, yes or no? Same thing is happening here. What did we do here is, we gave substrate. Initially what happened, enzymes were able to take the substrate and the reaction was happening faster and faster. Reaction was increasing. Can you see? Reaction was increasing. But after a certain point, where you cannot eat biryani anymore, at this point here, how much ever you increase substrate, how much ever I give you biryani also, how much ever if I increase substrate, see I'm increasing substrate. I'm increasing substrate everywhere. How much ever I increase the substrate, will the reaction go any faster? Not possible. Why? Enzymes are filled up. After a point, if I give you how much of biryani also, you will not be able to eat it. Why? Because you are done. You're done. You cannot eat anymore. That is called a saturation point. The saturation point is obtained here. So how does the substrate concentration vary, students? Substrate concentration, initially it will increase, but after some point it will become saturated. That is the substrate concentration. Same thing also applies to enzyme concentration also. Initially what happens? More enzymes, more reaction. But after some time, the substrate is less, Enzyme is more, so there's no point. Okay. Now here, if you take a graph here, if you want to study this entire thing, like at one what point your stomach became full? If I want to make a study, at how many plate your stomach became full? Like how much of chicken did you eat? Did you eat 2 pieces, 4 pieces? If I want to study all that, I need to make a graph here. This is the graph here. Okay. Now in this graph, I have something called as Vmax, half Vmax, and Km. Understand these terminologies. Understand. Now first of all, what is Vmax? Understand what is Vmax first of all. What is Vmax? It is the maximum velocity at which all active sites are filled. Maximum velocity at which all active sites are filled. It is like, at what speed did your stomach become full? Was it after a half biryani, 1 biryani, 2 biryani, 3 biryani? At what point your stomach became completely full? That is Vmax, maximum velocity at which all active sites are filled. Now students, understand, this is a little difficult to measure. Vmax is difficult to measure. So what do? Instead of finding Vmax, we find half Vmax. Okay, we find half Vmax. Half Vmax. Now what is half Vmax? Understand, velocity at which half of the active sites are filled. Half of the active sites are filled. That is called as half Vmax. Half Vmax. Now students, at this half Vmax, this is the velocity I got to know. Let's take after 10 minutes you became completely full. 10 minutes only you became full. Okay. In that 10 minutes, how much did you eat? If I want to know. See, at half Vmax, let's say, at half an hour, your stomach was full. You ate half an hour biryani and your stomach was completely full. But in that half an hour, how much of biryani you ate? At this half Vmax, I know that half of the active sites are filled. In this half Vmax, how much substrate is used? How much biryani have you eaten? How much substrate is used? That is called as Km value, Michaelis constant. Okay, that is called as Km value. Km value, Michaelis constant. Okay, what is Km value? Concentration of substrate at half Vmax. And remember students, always remember, okay, a good enzyme will always have less Km value because less substrate which it took less substrate and work also is done. So a good enzyme will always have less Km value because they're taking less substrate to reach the Km value. Okay, remember that. This is the Km value. Now students, we go to the next concept called as inhibitors. Can we start inhibitors now? Inhibitors also control the rate of reaction. They can increase the reaction or decrease the reaction. Can we start, all of you? Can we start? Can we start? Yes. Okay. Inhibitors. What are inhibitors first of all? Understand. Chemicals that shut off the enzyme activity. Inhibitors are the substances which will completely stop the enzymatic reaction. They are like, wait a minute, I am not going to let the enzymatic reaction work. I will stop the enzymatic reaction completely. You are not allowed to do the chemical reaction anymore. That is called as inhibitor. They are inhibiting. Okay. Now there are three main types. There are three main types of inhibitors: competitive, non-competitive, and uncompetitive. Now you can learn this all three different types with the help of a small animation which I'll teach you right now. After that, you will not have any doubt whatsoever. Okay, let me show you the animation. Now, first when you see competitor, now, see the animation first. Okay, so what happened here in the animation, students? This green one is the enzyme. Green one is the enzyme. Instead of enzyme binding here, instead of enzyme binding to the active site, who is binding? Who is binding? Red color one. Who is the red color one? Red color one is the inhibitor. So what is happening here is, in the case of competitive inhibitor, okay, competitive inhibitors resemble the substrate. This red color one is just like the green color one. So what will happen? Instead of green coming and joining, what is happening? Inhibitor is coming and joining, and it is becoming competitive inhibition. So as soon as the red color is coming and joining, the reaction is not happening at all. Reaction is not happening at all. Okay, competes with the substrate for the active site. So this inhibitor is fighting for the same active site compared to the, this is your enzyme. So enzyme, substrate is not able to join only now. Okay, see here, substrate is as such, substrate is not able to join, inhibitor is joining. So substrate is left separately, this is substrate. Okay, so what happened here? In this case, what is happening? Substrate is not binding. Substrate is not binding. Instead of substrate, what is binding, students? Inhibitor. Inhibitor is binding to the active site. Inhibitor is binding to the active site. Now, since inhibitor is binding, what do we get? We get inhibitor enzyme complex. What do we get? We get inhibitor enzyme complex. Substrate is not binding. Substrate is left as such only. It is left as such, substrate. Okay, that is a competitive inhibitor. Competitive inhibitor. Okay, the example of called competitive inhibitor is given in your NCERT, all students. Can anyone tell me the example? Can anyone tell me the example given in your NCERT, students? Anyone? The example of competitive inhibitor? Anyone? For example, succinate usually gets converted to fumarate. Usually gets converted to fumarate. Okay, fumarate. Okay. Now this reaction is possible because of succinate dehydrogenase. Because of succinate dehydrogenase is the enzyme here. It is the enzyme here. Now what is happening here is, there is a, this is substrate. Now just like succinate, there is a one more inhibitor called as malonate. Now what is happening? This malonate is now binding with the enzyme. Malonate is binding with the enzyme, and succinate is not binding. This is called as competitive inhibitor. So inhibitor is binding to the enzyme. Substrate is not able to bind. Okay, that is called as competitive inhibitor. Okay. Next one you see. What is non-competitive? All of us just see this. Inhibitor, substrate. Inhibitor is binding, changing the shape. Substrate is not able to bind. Non-competitive. It is called as non-competitive. Inhibitor is coming. It is binding other than the active site, changing the active site. This is not able to bind now. So binds at the site other than the active site. Binding to the site other than the active site. Changes the shape of the active site such that substrate is not able to bind now. So once the inhibitor is coming in binding to the other side, shape is changing, substrate is not able to join now. That is called as non-competitive. There is no direct competition. Behind the back it is going in the Katappa type. Katappa style, this is Katappa style. Okay. Can bind to free enzyme or enzyme separate complex. Both we can bind. It can bind to the enzyme, free enzyme also, or the complex also. Doesn't matter. Last one is called as uncompetitive. In uncompetitive, what happens? See this. See this. See, initially it cannot bind. Once the complex formation happens, then it will bind, it will kick it out. Understanding uncompetitive, what happens? In the case of uncompetitive, inhibitor cannot bind initially. Not possible. Not possible. So what will happen? It will wait. As soon as I join, then it will join and kick it out. Okay. It binds to the enzyme at site other than active site. Same thing. But it cannot bind to the free enzyme. It cannot bind to the free enzyme. It will wait. It will wait for the enzyme substrate complex formation to happen. Once the complex chain is formed, then behind the back it will come and bind to it. Okay. Done. We learned this entire inhibitor. Students, remember this one line here. This one. Such competitive inhibitors are often used in control of bacterial pathogens also. One line here. P by Q, that's why. Example is given here. Succinate dehydrogenase by malonate. Okay. This is optimum pH, optimum temperature at which the entire reaction happens. This is the entire Vmax, half Vmax and Km value. Small question here, answer in the chat all of you. What is the answer? Malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of succinate dehydrogenase. Now students, there are different types of enzymes, different types of enzymes. For example, there is oxidoreductase enzyme or dehydrogenase enzyme, right? Enzyme which catalyze the oxidoreduction between two substrate. Oxidation reduction happens. Transferase enzyme, enzyme catalyzing the transfer of group, that is Sg plus S. Now what will happen here? This G will be removed out. Transfer is happening. G is removed out. Hydrolase, enzyme catalyzing hydrolase of ester, ether or peptide bond. Okay, hydrolase happens. Then there is lyase enzyme which will help in joining. Okay, isomerase, all of you know, we use in glycolysis, isomerase reaction. Ligase is joining, sorry, ligase is joining. This is lyase, right? Lyase is removal, sorry. Lyase is removal. When there is removal, there is a double bond formation. Ligase is joining. Ligase is joining. Student, this you don't worry. Not many questions will come from here. You don't have to worry about this. What you need to worry about is this part here. Okay, this part here. This is the last topic in this chapter. Okay, that is types of enzyme and what is a cofactor. Now, what is a cofactor? Different type of cofactor. All that you learn in the next 5 minutes. I'll teach you what is apoenzyme, what is the holoenzyme, what is prosthetic group, metal ion, everything in the next 5 minutes, and the chapter is over. Okay, can we start? The last stretch. Last stretch. Can we start, students? Can we start? If you watched the video till now, if you watched the lecture till now, after the class is over, let me know in the comment section how was today's class. Well, it's a very easy chapter. I don't know why it's so difficult for many students, but it's a very, very easy chapter. After my class, just try reading NCERT once. The entire NCERT becomes so easy, I'm very sure. Very sure. Okay. Can we start now? The last stretch. Yes? No. Let's start, students. Now, what is enzyme? There are two types of enzymes. One is simple enzyme. Simple enzyme is made up of only protein, trypsin, pepsin. Only protein, simple enzyme. Then there is something called as conjugate enzymes. What are conjugate enzymes, students? Conjugate enzymes has a protein part as well as non-protein part. Both are present in the case of conjugate enzyme. I'll repeat again. In the case of simple enzyme, only protein. Conjugate enzyme, protein, non-protein part. Protein part is called as apoenzyme. Non-protein part is called as cofactor. Is that difference clear to all of you? Clear. Now this apoenzyme protein part is biologically inactive. It is inactive. Will it become active? It will become active only when cofactor is joining. When cofactor is joining, then it will become active. See here. Now, if the cofactor activates catalytic activity, okay, once they join together, that is apoenzyme, protein part, cofactor, when they join together, they become holoenzyme. That is apoenzyme plus cofactor if they join together, okay, and complete and biologically active. I'll repeat again, listen to me very carefully. There is something called as apoenzyme, which is inactive, protein part, non-protein part is called as your cofactor. This cofactor, right, this cofactor will activate the apoenzyme. See here. So apoenzyme plus cofactor is called as holoenzyme. And then it becomes completely active. Now there are different types of cofactors. Students. Okay, the three main types. Understand. First one is called as prosthetic group. Okay. Alright, here only. First one is called as prosthetic group. Second one is called as coenzyme. And third one is called as metal ion. Okay. Three main types of coenzymes, cofactors. Okay. Now, what is prosthetic group? Prosthetic group is mostly organic in nature. This also is organic in nature. This is inorganic in nature. Okay, inorganic. This cofactor will tightly bound to apoenzyme. This one will be very loosely bound. Or I can say only transient bound. Means very short time. Transient bound. Okay. Now example you understand here. Hydrogen peroxide will get converted to water plus oxygen. Okay. In this entire reaction, there are enzymes here. Obviously, there should be enzyme. Okay, let's take an enzyme peroxidase. We can also take, okay, peroxide oxidase. Now, in this enzyme, the prosthetic group I'm talking here is going to be heme group. Heme group is going to be the prosthetic group. So, example for prosthetic group is heme here. Heme is the prosthetic group. For peroxidase is the enzyme, apoenzyme, and this is the entire reaction. Now here example for coenzyme is going to be NAD and NADP. That is nicotinamide adenine dinucleotide. Now here they also have something called as vitamins. Example is niacin. Is the vitamin example. Okay. So NAD and NADPH are the coenzymes here. Coenzymes are the example. Okay. Now metal ions. Metal ions basically form the coordination bond. With the active site and of the enzyme and the substrate. Okay. And with the active site and the substrate, the example given your NCERT is the zinc. Zinc is a type of metal ion cofactor in the enzyme. Zinc is the example here, and the zinc is the cofactor here. Okay, in the enzyme carboxypeptidase. Carboxypeptidase is the enzyme, apoenzyme. This is the cofactor present there. Cofactors as well as the cofactors. Cofactors are non-protein part. They attach with the protein part, that is the enzyme, together making the holoenzyme. Okay. It is given here also in NCERT. Same thing which I have told you is given everywhere here. You can read the entire thing. You can read the entire thing once and you will be perfect in it. Now students, all the students who are watching till the very end, congratulations. You have truly finished this chapter. If you read the NCERT once and if you have any doubt after that, you can let me know the doubts in the comment section. I will clear the doubts myself in the comment section. And also let me know in the chat let me know in the comment section how was the class. And if you want this PDF, the PDF will be available in the description today only. That is my guarantee. Today only in the description the PDF will be available to you. Okay, so download the PDF, download the notes and be happy. Okay, and thank you so much students. Thank you so much. And I will see you in the next lecture. Next lecture is going to be this week itself. A special lecture is coming from my side. And let's see what's going to be done. Okay, so thank you so much students. I'll see you in the next video. Bye bye, take care, keep studying and smash the like button. Let me know in the comment section how was the video. Okay, bye bye. Boy.